My name is Gustavo Fernandes Mariano. I'm a PhD student from Brazil, and I'm here to show you my work called Evaluation of the Influence of the Environmental and Nutritional Factors in the In Vitro Cultivation of Pau brasilianata. So, Pau brasilianata is one of thousands species of tree in my country, and this is very special because we have the name of my country based in this species, Pau Brasil and Chinata. So it is most found in the Atlantic forest. Unfortunately, only half the coast of Brazil. I study Pau Brazil since my MSc student class in 2022, and at this year we have all the costs of Brazil with Pau Brasilia, and now, 4 years later, only half that I said. So this is because the extinction of the species basis of the illegal economy, the illegal traffic, because the wood used for make musicians' instruments, and also the color red of the wood used by paint, fabric, and clothes. So this caused all the extinction of the pitches. So we make a lot of study in vitro cultivation to save the tree and not be extinct. So we have in tissue culture in vitro a lot of techniques and new students. So we make photoautotrophy and photomixotrophy to test it in these pitches. So we thought about photoautotrophic. We intends when you use the medium MES and the medium with MES and sucrose, but we separate. When I have sucrose in the compositions, we call photoautotrophic. When we have a sucrose, we call photomixotrophic. So we thought, let's try these two types with 4 different type of sealing on the flask. We use it the PVC, polychlorate of vanilla. The plastic using to protect the food, put the fridge, is the same. Simple cap, a cap with one-membered microporous members that allowed gas exchange, and another with two microporous membranes. So with this, we thought we're gonna test photoautotrophic and photomixotrophic. We're gonna test different type of sealing. Why not test the quantity and quality of lights too? Because we know that we have the red lights with 700 nanometers and the blue lights with 400 nanometers. So when we put this together, we have the pink light. So these two colors important for the photosynthesis, chlorophyll, anyway. So the pink lights maybe is the best. We'll see. So this is the process. We take the seeds. I open. I forget the name, but I call case. And this is a photo of zygote embryos. And this is the Brazil in vitro without contamination. So we thought about photoautotrophic cultivation promotes a better development of Brazilianata when use microporous membranes. Will gas exchange provides more efficient absorption of water and nutrients to make a stomata functional. The intensity and quality of the lights with happy activation of the photosystem and increase the carbon fixation. And will the results by evidence by superior morphological values compared to photomicrotrophic cultivation? We know. We're gonna see now. So this is the results of 19 days of in vitro cultivation. And this, when we start to get crazy, because we can see beautiful plants, we live with roots, but not every this one make it happy that is good for the plants. Look this. So we have pink lights with sucrose, pink lights with no sucrose, white lights with sucrose, white lights with no sucrose, white and pink lights with sucrose, white and pink lights with no sucrose. So after this, we make some tests. Here we evaluation of the length of the aerial part. So this is only for the pink light, okay? Only pink lights. So the plants in pink lights with no sucrose, we have the best values for the two members. We have the most part of area, um, not make some difference here, but it's for pink lights. Here we have the pink lights with sucrose. So we can see that the other type of ceiling was better than we see the two members. That's why, because have gas exchange, so the functional photosynthesis, but here we not good results. That's because when you have sucrose and the photosynthesis that make maybe well, we don't know, we can stop the plant to absorb nutrients from the plants because we stop the limit, the wave of the water. So this stop the plant to absorb correctly the nutrients. This is for the white lights. Then we can see when no sucrose, the two members again, the bad values, the good plant, and we have sucrose again. With two members, not good for the plant, maybe it's not valuable. This for the white lights, and this for white and pink lights. Again, when no sucrose, two members is good, okay, photosynthesis, no accumulator of sucrose, and this when I have a sucrose again, two members, no good for the plants. So beyond the aerial part, we saw two roots lengths. So this with no sucrose, again, two members is good values, a good plant, but with sucrose again, the others was better than two members. And here, only the number of the leaves, we call each of one this, we have a one membranous with the best results. And here we have only between the medium with MES with no sucrose and medium with MES with sucrose. There we can see with no sucrose, again, two members were the best, good photosynthesis, he can make it herself, but with sucrose again, no better results. So it is. Before I finish, when I make these abstracts for this conference, we don't have the whole results. So we can say right now it's good using two members for the plant with no sucrose. That's good because I don't have to pay sucrose. So it's economy for my laboratory. But it's not working with all the species. I have tested with 3 species, Pau brasilianata, Cariana legalis, and a species of coffee from Brazil. And each have one response difference. But between the lights, Pau Brazil is better in white lights because it's plants for the tropical country is heat, so it is good. Cariana is the best lights with pink. She don't like to be so heat, so warm. And coffee also was pink and white lights. So each species is different for what you see. And what I can say now that I finished, that I have 2 members and no sucrose were the best for Pau Brazil because when I take from in vitro to ex vitro to survive in nature, 2 members and no sucrose were the best. So I have more number of plants successfully on the nature. So that's it. I want to say thank you for Institute, thank you for my lab, my university, and you guys for having me today.
Thank you for this interesting presentation. I might say that pink light is quite exotic for me. And now, please welcome our next speaker. And our next guest came from Warsaw. Please welcome Antonina Dubańska. I think that we will listen to a presentation about rock resistance of beech and oak seedlings. So we stay with seedlings. The floor is yours.
Good morning. For today, I prepared a presentation about morphological and recovery potential of 2 very important thank you. for two very important forest-forming species for European temperate zone, so Fagus sylvatica and Quercus robur, under water deficit and soil drought condition. So drought-related stress is a very important factor for research about adaptation of forests to the climate. We already observe that drought is accused of the increased the mortality of the trees in Europe. Not only in Europe, but I will focus about this region, and that these very extreme events, they are more frequently observed. So the recovery time possibly for trees is also decreased. And this influence on the potential resilience of our trees and the whole stands. So what we can learn from the climatic models is that this trend will be continue in the future, and we could observe the shifts of the potential ranges of our species. So this is very important for us now to deepen our understanding of how trees respond to water stress. Since the plants start to colonize the land from the water, they have to learn how to cope with water limitation condition. Today we can divide them into two groups due to their behavior, to the isohydric and anisohydric type of the behavior. For isohydric species, we can say that they have more sensitive stomatal regulation. So if they have to cope with water limitation condition, they close stomata early during these conditions. Anisohydric species, on the other hand, they have less stomatal sensitivity regulation, so they try to maintain the gas exchange for a longer, and they just try to keep going and don't look back that, oh, this is the drought, so I'm sorry. So this is two types of the behavior, but it's not very simple to say which species is oak is typical anisohydric species, whereas spruce we can say is more isohydric species. But the beech is depending on the provenance and on the research. Some results they show that it's more isohydric or more anisohydric species. So these have some features from in between these two types of the behavior. These both behaviors, they have some advantages and disadvantages, and they are also related with some features of the species. Also from the morphology side, which was very interesting for me. This is why the aim of my research was to identify and compare the effect of water deficit and soil drought, so the same stress but with different intensity on the morphological changes in the leaf, shoot, and root system, which is much rare considered in the research of these two species, as well to assess the specific capacity for the recovery, because the recovery is this factor that is very important for possibly the species to survive during this more frequently stress condition. So what we do, we provide the experiment under controlled irrigation conditions to maintain the forest microclimate we put our seedlings in the forest gap in the central Poland forest in the central Poland. We put the seedlings under transparent roof to avoid the precipitation and on the plastic performant panels to avoid the soak of the water from the ground. For optimal irrigation conditions, we consider the level of 0.4 square millimeter of water per square meter of the soil. For the water deficit, it was 50% of the optimal irrigation dose, and during this water deficit, the amount of the water decreased to 0.2. And for the drought, we just suspension the irrigation temporary for 30 days, and the level of water decreased below the 0.1. So this is the level that if we touch the soil, it will be completely dry. So we provide the water directly by the pipe system to each pot separately. And we were measuring the water content in the each pot during the experiment. For the destructive measurements, we measured 220 seedlings, which were collected from the 3 terms of the experiment. First, it was directly after stress. Second was after short recovery time during the same growing season. And the third time, it was after 1 year of the recovery in the second year of growth of the seedlings. For the non-destructive measurements, we measure over 1,000 of the seedlings for the measurement of the height and the root collar diameter. So this destructive measurements, we collect material from the leaf, shoot, and roots separately. We dry this biomass and weight it, but before that, we also scan the leaf area and the roots with the scanner and WinRiso software to provide more detailed information about the root architecture. So for the results, in the first slide, we can see this big difference between the severe drought, and the water deficit and the control. We see that during between the water deficit and the control, there was no significant differences between both the height and the root collar diameter, but there was an important difference between them and the severe drought, and these differences were also maintained after the long recovery time. So still the seedlings were significant smaller than from the water deficit and control treatment. For the biomass, we didn't catch the differences in the leaf biomass in the beech seedlings, but we see the differences in the shoot and the root biomass, especially during this directly after stress measurements. But what is very interesting here is that this water deficit was the biomass there was even higher than from the control seedlings. So this water deficit make this growth of the seedlings even better for the shoot and root biomass. Similar we can see in the oak seedlings, but here we also catch that the leaf biomass in the severe drought decrease compared to the water deficit and the control. And also we can see here the differences for the root biomass. From the scanning of the root architecture, we catch the differences between the drought and water deficit. But what was surprising for me that we didn't see the differences between the severe drought and the control seedlings. And the same it was for Fagus sylvatica as for the Quercus robur. But for Fagus sylvatica, we see that these differences were also visible after this 1 year of recovery. But in the oak, these differences were only after this stress, and then it was no significant differences. I also calculate the PCA. That we see that these clusters, they are less concentrated in the Fagus sylvatica. So these differences between treatments was more visible in this species, which is also important for this recovery period. Whereas during in the Quercus robur, these treatments are more concentrated, so the differences between treatments were smaller. To conclude my research is that the drought significantly reduced the high and the root collar diameter growth in the both species, but the water deficit have no visible effect on the growth, but stimulate the undergrowth biomass and surface area of the roots, and can induce a kind of the compensatory mechanism that differ from those that we observed during the severe drought condition. Comparing the species, we see clearly that oak, as a more typically anisohydric species, show a greater stability and maintains maintained during the soil limitation conditions compared to this less anisohydric beech seedlings. For the recovery, the most important result is that the oak was able to fully recover during even the same growing season, which was much faster than for the beech seedlings. And after the recovery, we see still differences between the growth, but no differences between the biomass allocation. Our study have some practical implication, especially for the trees nurseries, where we can see that the water deficit could be beneficial for maintain this better adaptation for water limitation. Thank you very much. This study wasn't published yet. I'm still working on the paper, so I especially encourage you to make some questions for me.
Okay, as you wish. Thank you very much for your really interesting presentation. Let's move to the next speaker. And now we welcome Joanna Kijowska-Oberts from Institute of Dendrobiology. And we'll also talk about drug, but in the context of the role of proline as a multifunctional compound. The floor is yours.
Thank you very much. Hello, everyone. I am Joanna Kijowska-Oberts, and I am a PhD student in the Institute of Dendrology PAS. In my presentation, I will try to explain if proline is a stress tolerance marker or a protective substance, or maybe both of them, and why. We often hear about climate change, and we know that it causes shifts in the distribution of many plant species. However, it is rarely mentioned that these changes in distribution often originate from seeds. High temperatures and water deficits, which are the effects of climate change, affect seed quality while they are still developing on parental tree. And such conditions induce stress, which can lead to a loss of seed viability and a decrease in germination ability. But on the other hand, stress can also induce acclimation and improve resilience to unfavorable environmental conditions. Populations acclimatized to new climate conditions can serve as a source of seeds that do not lose viability during long-term storage. This kind of storage is highly needed in forestry and nursery production. Therefore, through identification of populations producing high-quality seeds, the economic losses related to decreased germination capacity could be reduced. The efficiency of seedlings production would increase, and these seedlings may be more tolerant because they would come from populations identified as better acclimatized to current conditions. But how can we identify such populations? Biochemical markers may be helpful. Proline and other chemical compounds related to its metabolism are promising indicators of seed and seedlings quality. Proline is a unique amino acid. It accumulates in cells in response to stress. Proline scavenges reactive oxygen species molecules, which are abundant under stress conditions and damage cellular membranes. It protects protein structures and stabilizes membranes building cell organelles, and it also prevents water loss from cells because of its osmoregulatory properties. Proline metabolism and its antioxidant properties are relatively well known, but until now they have been studied in herbaceous species such as Arabidopsis, maize, or rice. However, the results of studies on short-lived species do not fully reflect the mechanisms occurring in seeds of long-lived plants. Therefore, we took a closer look at 2 tree species, Norway maple and sycamore maple, because on the one hand they are closely related, but at the same time each of them produce seeds belonging to different category on the basis of desiccation tolerance. So Norway maple produce orthodoxies, which are resistant to water loss, and sycamore produce sensitive seeds, so they belong to recalcitrant category. And based on studies conducted over 3 years, we demonstrate that proline content is dependent on moisture and thermal conditions in recalcitrant seeds. Because we noticed correlations of proline accumulation with mean temperature and mean precipitation in seeds of sycamore maple. We therefore decided to go a step further and compare the strength of relationship between proline accumulation and drought stress of seedlings from different tree species. We conducted the first meta-analysis covering the role of proline under stress conditions, while also focusing on the seeds and seedlings of trees. And the size of the effect, which is accumulation of proline under drought stress, depends on the duration of drought. As you can see, the optimum accumulation of proline occurs after approximately 70 days of drought, while this effect is non-linear. And what could this turning point mean? Probably a longer period of drought leads to a state where the level of drought stress is so high that antioxidant system cannot effectively remove reactive oxygen species. But we have one alternative theory that it could be the moment when acclimation occurs and seedlings stop to experience drought severely. Another model shows that the effect size depends on seed category, and there may be a relationship between seed category and ability of seedlings germinated from them to tolerate drought stress. We have also demonstrated that the effect size differs between deciduous and coniferous trees. And moreover, the greater seed mass, the higher proline accumulation in response to drought. Besides proline, enzymes involved in its metabolism are interesting potential markers of drought tolerance. Proline biosynthesis in plants occurs in cytoplasm, and it's catalyzed by pyrrolene 5-carboxylate synthetase, P5CS. Under drought and osmotic stress conditions, the dominant substrate is glutamate. The activity of P5CS is directly stimulated by ROS, including hydrogen peroxide, whose levels increases in response to stress. And proline catabolism takes place in the mitochondria, and it's catalyzed by proline dehydrogenase, ProDH. ProDH activity is inhibited under drought stress conditions, but induced by proline accumulation. To investigate changes in proline metabolism in seedlings of tree species during drought stress, we used seedlings of Paulownia tomentosa. We exposed 1-month-old Paulownia seedlings to varying moisture levels. The research material, divided into leaves and roots, was then collected 3 times at 10 days intervals. And what did we find out? The increase in P5CS activity was correlated with the increase in hydrogen peroxide accumulation. We expected that result. But what is very interesting for us, the strongest correlation was observed between ProDH activity and soil moisture level. And we can see this strong correlation here and here and here and here and here and here. So in every collection date, in leaves and in roots. And this suggests that the activity of these enzymes, particularly ProDH, may be more reliable indicators of stress than the proline concentration itself. This approach could find practical application in forestry because seedlings identified as experiencing lower stress levels are likely to develop into forest stands with greater resilience to changing environmental conditions, especially to reduced precipitation, of course. Moreover, we noticed tissue specificity. P5CS activity was higher in roots than in leaves, unlike ProDH activity. So on this basis, we assume that roots are the main site of proline biosynthesis and leaves are the main site of its catabolism. All right. So to directly address the question posed in the title of this presentation, I believe that proline can serve as a marker of drought stress tolerance precisely because it simultaneously acts as a protective substance that prevents plant cell from damaging effects of drought stress. And I hope that in future perspective, markers related to proline metabolism, such as ProDH, will help us to identify seedlings which are more stress resistant and to increase the resistance of subsequent generations in forestries to new climatic conditions. We really want to manage forests in a way that makes them more stable, but we will see. But that is still ahead of us. And what about now? Now I am conducting research within a grant I received from the National Science Center in Poland in the Preludium 22 call. You can see here the title of my project. I am comparing seeds of Scots pine from 3 different climatic regions of Poland. And among other things, in terms of oxidative changes and proline metabolism, we obtained seeds from 3 different locations, one from each climatic region. And now I will present the preliminary results. The level of proline and hydrogen peroxide are the highest in seeds from this location. It is a location with, which is characterized by the lowest temperatures and the highest precipitations. And you will have to take my word for it. The germination capacity of seeds from this location was the lowest. So I think that is because the tolerance to environmental conditions changes in seeds from this location, this is the same location too, the tolerance of these seeds is lower, so they accumulate more proline because they experience the higher stress. However, I will soon start measuring enzyme activities, and I am curious to see what they will reveal. Thank you very much for your attention.
Thank you very much. In your last sentence, you asked my question, so thank you. And now we stay with the Institute of Dendrobiology, but we'll change our topic a bit. Now we'll listen to a presentation of Elvira Nawrocka about micropropagation. Elvira, the floor is yours.
Good morning, everyone. As Professor Szuba said, I'm Elvira Nawrocka, and I am doing PhD at Institute of Dendrology. And I will tell you something more today about maybe not our results, because we still work on them, but mostly about interesting technologies that we used in our research. So why even to study Picea abies, which is Norway spruce? It is ecologically important. It has ecological importance, as you know. It is key boreal and montane forest species, and it has biotechnological relevance. It is useful for clonal propagation and improvement, and the core problem of our study was tissue survival varies widely. And what challenges facing Picea abies in nature? As you know, over the world we have problem right now with drought and climate change, and increased temperatures and reduced rainfall lead to severe water deficits, weakening trees and making them more vulnerable, for example, for pests and for diseases. And outbreaks of bark beetles and fungal pathogens cause widespread mortality exacerbated by stress. We have also problems with environment, as I said before. So also drought is also an environmental stressor, but we have also problems with air pollution, acid rains, and nutrient imbalances, as you can hear before about nitrogen, for example, and what further degrade forest health and resilience. Also, important is habitat loss and proper habitat management. And unsustainable forest practices, clear cutting and fragmented habitats reduce genetic diversity and adaptation potential, what in the future can conclude into pests and diseases and other problems of those trees. And understanding tissue culture and viability is crucial for developing resilient strains and implementing conservation strategies for this ecologically important species. Our objectives of this study was what we was doing with embryogenic tissues of Physea abies. We had 13 lines, genotypic lines, and we wanted to assess its survival. So we need to compare survival across those selected lines. Our aim is to find genotype-specific indicators of those lines' viability and build a profile to combine key physiological signals linked to survival. Our analytical approaches were three, like hydrogen peroxide quantification, which indicates oxidative status. Seahorse analysis was showing us metabolic activity, and also ultra-high performance liquid chromatography with tandem mass spectrometry was helping us with show regulatory signaling. And as you probably know, this hydrogen peroxide, it is a key reactive oxygen species used as an indicator of cellular oxidative status, and its quantification allows us assessment of oxidative stress intensity in plant tissues. Here you can see that dynamics in Physea abies of hydrogen peroxide was in most genotypes time-dependent and it was increased, what indicates a progressive accumulation of oxidative stress during culture. And response is strongly genotype-specific with clear differences in baseline levels and accumulation rates. And you can also see, maybe not that good on this graph, but you can see that some of lines didn't even survive to second stage. So this is really important to see what's the problem with them and how to care properly for them, to have them longer in those culture, because if you want this specific genotype, we need to see what's the problem and resolve it. And the next methodology that we used was oxygen consumption rate, which is a measure of cellular respiration that quantifies the rate at which oxygen is used by cells, serving as an indicator of mitochondrial activity and overall metabolic function. And it is a really useful technology that I just know in the institute, because although OCR is most commonly applied in animal systems, for example, cell cultures analyzed with seahorse technology, it can also be adapted, and it is in our institute, for plant tissues to assess respiratory activity under different physiological or stress conditions, just as it was used for animals' tissue cultures for something And here you have nice diagram of what we was doing with those embryogenic tissues. And it first was, of course, sample preparation. And maybe intact isn't that proper word for it, because we needed to separate those cells from each other. But we want them alive, of course, because they need to use oxygen and everything. So like next we was using cartridge that are specific for this model. And later we had measurement with special oxygen sensors. And later we have, for third stage, we have data acquisition and, of course, data analysis and interpretations. And here, because we have so many lines, we have also many results, and we still need to get into it and take more care of what we want to analyze and everything. So the first sampling term revealed strong genotype-dependent differences in OCR, indicating substantial variation in mitochondrial respiratory activity among embryogenic spruce lines. And OCR dynamics were highly non-uniform over time, suggesting genotype-specific regulation of energy metabolism and differing levels of metabolic plasticity. The third sampling term revealed strong genotype-dependent heterogeneity in OCR, and overall OCR dynamics were highly variable and non-linear, indicating substantial metabolic plasticity and unstable mitochondrial regulatory in embryogenic tissues. But as I said, these results are still in calculations and everything, so keep it in mind. And our fifth term sampling shows a general stabilization of OCR dynamics compared to earlier time points, although clear genotype-dependent differences persist. For example, here we can see that SC2 lines display distinct respiratory profiles, with some genotypes maintaining consistently higher oxygen consumption rates, while others exhibit lower and more stable respiration. But as I said before, you need to keep in mind that some of lines didn't survive to the last stage of sampling, so yeah, we have still problem with it. And the methodology that we used also, which is really interesting, and you can use this in your research if you have opportunity, I can recommend, because ultra high performance liquid chromatography with tandem mass spectrometry is an analytical technique combining chromatography separation and mass spectrometric detection. And it is used to separate, identify, and quantify compounds in complex biological samples. And it works in 2 steps, as you can see here. And here you have also a nice diagram of how it works in stages. And this diagram illustrates this workflow of this methodology. And first, compounds are extracted and separated by chromatography. And then they are ionized and analyzed in the mass spectrometer, where precursor ions are selected and fragmented. And finally, data are used for compound identification and quantification. And this comprehensive approach ensures us of high accuracy and sensitivity. And this is what you need to keep in mind also, because this is a really sensitive procedure and technology. So if you have any contamination, even not of your fault, the program can read automatically some peaks as your standard or your samples, and it can be totally different thing. So even if you want to automatically do it, you need to later check every chromatogram by yourself. And if you have many samples, it's a bigger problem. And yeah, we have many lines, and this is really early results. So keep keep it in mind. And no consistent temporal trend in alleloacetic acid levels were observed across genotypes, and it's indicating non-linear genotype-dependent hormonal response. And it is strong genotype and time interaction suggests that auxin dynamics are mainly driven by genetic background rather than developmental stage. So yeah, we need to work still on those results. Here I chose the most stable lines, but those still indicating stronger hormonal homeostasis and reduced temporal variability. And abscisic acid showed a relatively stable overall profile, and with this we are more okay and fine with those results, but across all genotypes and sampling terms, there was no consistent temporal trend still, but variation was primarily genotype specific rather than time dependent, and it indicates a generally buffered hormonal regulation. And here also I chose 5 the most stable lines, and also as before you can see that many of those lines didn't survive. Here, yeah, from 13 we have 10 at the end. So future perspectives and conclusions. Conclusions are genotype-specific survival patterns. So we have hydrogen peroxide OCR and those 2 hormonal regulators. And those profiles reveal strong genotype-dependent survival responses, and 4 lines didn't survive cultural conditions at some stages. Redox balance drivers differs by line. SC2 lines showed elevated H2O2 levels, indicating stress-related processes associated with differences in tissue viability. And next, metabolic plasticity, OCR patterns varied by genotype, reflecting distinct respiratory and energy management strategies. And hormonal regulation, IAA exhibited high genotype-dependent plasticity, while ABA remained relatively stable, suggesting differential stress response regulation. And our future perspectives are that these physiological markers can serve as early predictors of cultural success prior to visible survival outcomes, but you need to care that embryogenic tissues are really difficult tissues to obtain, to do some analysis of them, even with I don't know, with grinding them, there was a problem when it was frozen. So at least even at this first stages, it was a problem. So overall, genotype-specific physiological profiling is crucial for optimizing cultural conditions and improving spruce line resilience. And thank you. For my research team also, Hanna Fuchs, Teresa Chazubska-Przybył, and Professor Ewelina Ratajczak, and acknowledgements to Professor Jacek Kęsy and Karol Lesiakowski for technical support. Thank you.
Thank you very much. Thank you very much for this really interesting presentation. For a moment, I thought that we have tandem MS in our institute, and I don't know about it. Okay, and now, last but not least, Adam Bąk from Poznań. And we'll stay with in vitro propagation, but this time we will listen to a presentation about oak rooting. Really also a challenging task. The floor is yours.
So, welcome everyone. My name is Adam Bąk, and I'm currently a master's student, which I'm finishing by University of Life Sciences in Poznań. Today I would like to talk about my master thesis with you, which which was been made in the Polish Academy of Sciences Institute of Dendrology with my associates. And the subject was morphophysiological factors regulating rooting of pedunculate oak, Quercus robur, in vitro. Why we focus on pedunculate oak? So let's start from the beginning. Quercus robur as a species play a major role in forest regeneration process due to its good resistance to frost and sudden climate changes. As an economically important species, there is a high demand for its woods that is used in carpentry and building industry. Demand for high-quality planting material is increasing. Unfortunately, current methods are not as good as we want them to be. Grafting is inefficient method of propagation with only 60 to 70% of success. As we should know, the acorns are recalcitrant seeds, unfortunately. This means we cannot properly store them in sub-zero conditions. Moreover, oak don't produce the same amount of seeds each year, which is a problem for us. Sometimes we have to wait 5 up to 10 years for an abundant yield of acorns, which stops the production of seedlings in forest nurseries. So that's where the in vitro part comes. In vitro can help with this challenge, since we can use every plant cell or organ to regenerate a whole plant out of it. As an important fact, in vitro as well allows us to clone the plants so we can get the saplings with desired genotypes from donor trees. The research problem we had was clonal propagation of Quercus robur is limited by low rooting efficiency and multiplication. Rooting of mature donor material was and is particularly difficult. Young explants perform much better in vitro cultures, and current protocols that were used by Institute using 1-naphthalene acetic acid and activated charcoal showed low effectiveness in rooting. Our hypothesis was indole-free butyric acid significantly enhances rooting efficiency, and metatopoline reduces physiological stress and improves plants' response to auxins. Why we chose IBA? As we all know, the auxin enhances rooting in plants. We chose IBA based on the research that was already made in our department. And why we chose metatopoline for this process? Metatopoline as a cytokinin boosts chlorophyll production, reduces stress in plants, and stimulates the formation of lateral buds and elongation of the stem. So basically, what we want to achieve in our study was how to make the explant that we see on the left photo go to the explant that we see on the right photo, which should be our final product. Plant material we used buds that were come from epikórnik shoots derived from a 90-year-old oak, which was a donor tree. It is located in Dąbrówka Kościelna in Poland. Before an experiment, the in vitro cultures were maintained on standard WPM medium supplemented by BAP, subcultured every 28 days. The growth conditions were 20 degrees Celsius, 16:8 photo period, 8 hours of dark, 16 hours of light, under photosynthetically active radiation. The main thing that appeared as a selection of an explant was their length and their callus at stem base. Experiment design, we collected total of 400 explants, which we derive into 2 groups of 200, and each of those 2 groups we derive into 4 groups of 50 explants, and the culture conditions. So basically we took 400 plants, then each group of 200 plants was incubated in IBA medium, which contained IBA or which didn't contain the IBA. Then each group was divided into in vitro and ex vitro part. In the in vitro part, we kept plants 40 days on the mediums with or without metatopolin. For the ex vitro part, before we put them into the soil, we soak them in the fungicide solution to prevent any unnecessary contaminations. Then they were transferred to substrate and they were growing 60 days in conditions showed on the screen. Every 7 days, the ex vitro plants were sprayed by a metatopolin solution or just with the plain water. Culture conditions, as you can see, on the left, the in vitro plants that were in the metatopolin medium, and on the right photo, the oaks that were put in the soil solution. Medium used in research, we used modified WPM medium. As I say modify, I mean we put more sucrose. Yeah, sorry for this. Wait a second.
Okay, I think that because of health issue, we'll end it now, and that's it. Please take a seat. So we'll break this presentation at this moment, and I think that we just, finish our session now. So it's a little bit surprised, but I think that we now have a lot of time for discussion, and I personally think that also this topic is a really challenging task to rethink oaks, and really I'm sorry that we cannot ask any question, but we have a lot of time for next speakers, and I will start a discussion now. I think that all presentations are really huge, super interesting, that I hope that there will be a lot of questions. So I will start with the questions to now our last speaker, to you. Do you have any questions to Talia? I know. To Elvira, I'm sorry. No, I'm really confused. To Elvira, of course. Do you have any question to Elvira? Okay, the first question is from Warsaw.
You were choosing these few stands for your research from different climatic region of the Poland, and how you specified the choose which stand it will be. It was on the same age. You consider the soil, the population. Yes. No, it wasn't.
I don't have any population of some stands. Okay, so I'm not sure. I don't know. Maybe I. Okay. Okay. So who has presentation with stands in populations of? Because it wasn't me.
Yes. I think that we are all a little bit confused because of the switch, but Elvira told us about the micropropagation of Pinus albius. And this was quite biochemical presentation. Do you have other questions for Elvira? So I just have maybe not the question, but a little bit anecdote. When you're talking about contamination, during one of my experiments, we detect a huge number of cannabinoids in poplar roots, and we was happy for several times, and we planned some little production maybe but it's really quickly realized that it's just contamination from the previous study. And my question is, how big the sample was for this oxygen consumption study? How many milligrams you need to analyze it?
100 milligrams, I'm not sure right now. So yeah, it was 100 milligrams, I think. so yeah, it's not a big amount of
Oh, for in vitro culture, it seems to be a lot. So, any further questions? Then we switch to Joanna Kijowska. Oh, I'm sorry, I didn't see.
About the measurements of OCR on seahorse, it's a high throughput method, so it doesn't require a lot of starting material to the measurement.
Yes, in my word, 100 milligrams is really a lot. Okay, now we switch to the Joanna Kijowska-Oberts, and I'm looking for the question to Joanna. Yeah, anyone? Because I had a question, but Joanna asked actually in her last sentence, because I just wanted to ask about hormones, enzymes. So it seems that we also go to the next speaker, and now I ask, yeah, how you do? Because I didn't see it.
I have, yeah, this was for you, and also I have a question, why Pawlownia? Because you said that this have implication for the forest, but I think not our forest, because Pawlownia is not from here, yes. Okay. And it's very specific as a metabolism.
But it's model tree species which is used to a lot of studies because, for example, because it grows very fast. Yes, so it's a model species and it's okay to use it.
You think it's good to compare this species with our native species?
Yes, why not? And you know, it's the first study on these enzymes on tree, and we will check it on another species.
Okay, and I think that now we just switching to the lady from Warsaw. Do we have some question about rock and things? Now we are question Lady Antonina. Sorry, Dubińska. I'm sorry.
You said at the end of your presentation, you said that water deficient can stimulate growth of undergrow biomass. I will ask if it's seen after the long period of time, or also after short period of time it was also seen?
It was more visible in the beech seedlings, but because in the oak this was only visible direct after the water stress.
Okay.
After recovery, these differences just disappear, and all the treatments were similar in the oak, but in the beech we still have some differences between them.
Okay. Thank you very much. You have answered my next question at the same time. So I will first ask about the period and then about the species. Thank you very much.
Okay, and I also ask for the next questions.
I have a question. What about a closer look to recovery, for example, on biochemical levels? And do you think, because your basis was morphological changes, and do you think it's enough, and do you have some plans to check it?
Yes, this is also interesting. The whole of my PhD is focusing on this three level: is a biochemical, physiological, and morphological. But this research is this from the morphological part of the experiment.
Okay, great, thank you.
Anyone? Oh, a lot of questions.
I have a question more like for practical forestry. You said that the oak is like the better resilience on the stress conditions, the water stress, yeah? So do you think that we separate them in forestry management? We don't grow together oak and the beech. So do you think that in the future management of forest we should check the microclimate and put oak seedlings where is the more stress conditions with water, and the beech seedlings where is less stress conditions with water?
Yes, but this is also from this preferation of the species. Beech is a species that prefer more fertile and more moisture size than oak, which have this broader range for the condition. I answer, or you did something more?
Yeah, you answered, yeah.
And maybe one question from me, or the other questions from the audience? It was a lot of questions, so maybe the last one, because I think that you want some proposition for the other analysis, and my question is, did you analyze the level of root colonization? Because you analyzed pot experiment and those plants growing in the middle of the forest almost, so I think that they were colonized, and mycorrhiza has a huge effect on tolerance, and you have roots, so it's possible to do. Do you think about analysis of mycorrhiza or root colonization?
I didn't check the mycorrhiza, but I know that this is also the important factor. But I just don't have a chance to do that.
Yeah, but you have roots, so you still could do it.
Now they are dry.
No, it's not important, I think.
Yes, it's still possible.
I think that you could use, for example, analysis of ectosterols, some sterols. Maybe it's possible. I don't know exactly if they are dry, but we have a specialist.
I didn't have enough time for the root system analysis during the presentation, but I also check the proportion of the smallest roots to the more thicker roots.
Yeah, exactly. So this was my suggestion, and now we could switch to our first speaker. Do we have a question to Gustavo? Yeah, any question for the first speaker?
Yes,
yeah.
Because you was showing us mostly the picture, and you said that this was better, this was less better. But do you have some quantification, like you use some statistic to say which is really better, not just by the look of the seedlings?
Yes. This is the next step, because we saw bigger roots, bigger area parts, but we don't know the stomach area, the photosynthesis inside the plants. So this is the next step to realize, say, okay, 2 members is good, high oxygen gas exchange is better, so don't need sucrose, is more cheap for your lab, economy. So this is we're gonna do it yet. We not do it yet.
Thank you. Do we have any further questions? Okay, thank you very much. And now I receive information that we'll be able to show the whole presentation that we film. Just the floor is yours the next time. So, please.
Oh, so welcome back again. My body wanted to stop me, but I didn't give up. Yeah. Now we're gonna have like a more proper presentation because I'm a student, not a scientist as you, so don't worry. As we're back, conditions of the plants, we talked this. Let's go back to the medium part. Okay, the mediums that you used were modified WPM mediums. Basically, the modification was we added more sucrose because the plants weren't keeping 28 days straight on the metatopoline medium, just 40 days, so the plant needed more sugar. We used ppm in both mediums, even for the proper grow and for the incubation. ppm is a plant preservative mixture. It prevents our in vitro cultures from getting contaminated by bacteria and fungi. For the rooting medium, we also used activated charcoal, which is an absorbent in medium. By absorbent, I mean when oak lives in vitro culture as a product of metabolism, he produces phenol, which is toxic for it. The main reason is for the charcoal is to absorb this phenol, so the plant won't be stressed. Plus, activated charcoal makes the medium dark, which enhances rooting of plants in vitro. For the mediums without IBA and without metatopoline, we just use exactly the same, but just without the addition of those two components in them. For analysis, we made some simple and more complex analysis. The simple ones were the plant fresh weight measurement, the root system analysis of length and number of roots in the plants. For more complex ones, we made photosynthetic pigment analysis, which means it was an analysis of chlorophyll A, B, and carotenoids. On the pictures, you can see the moment I was measuring the weight of an eggplant, and in the left picture there is like a grinder plate with the analysis of pigment. We also did chlorophyll fluorescence analysis, the analysis of photosystem second by the Pocket P Hansatech device, and anatomical analysis, which was a cross-section of leaf, stem, and root. For leaf, we were measuring the spongy, palisade parenchyma, and stomatas. In the roots, we measured vessels, and in the stems, we measured vessels as well. So on the photos, you can see actually the microscope and the cross-section of leaf of the oak. And in the left photos, you can see the moment of analysis by the device I mentioned earlier. It has to be the green light because the plants are needed to measure in the morning when they are still in the dark photo period. The green light is not a light that is photosynthetically active, so we use the green light so we can see anything what we are doing there. That's why the light is green. So now for some results. Those are the Petri dishes containing each 3 explants coming from each treatment. The lower row is an ex vitro plants, the upper row is in vitro plants, and the IBA minus are the treatments without IBA, and the minus MT are the treatments without the metatopoline. As we can see, we have 2 Petri dishes are much different from the rest. The main difference we can see from the upper row and the lower row is the size of an explant. Their ex vitro parts are much smaller. I mean, the stems are shorter, much thinner than the top ones, and the roots are much thinner, shorter, and there are no more than 1, maximum 2 roots. In the in vitro, however, we can see that those 2 Petri dishes that comes from the culture that was in the IBA have strong roots, which are thick and long and more than 1 root we can see, and they have a lot of secondary roots. And the condition of plants are better, the leaves are bigger and greener. Those 2 tables are showing us the percentage of the plants that are rooted. we were expecting some percentages, like for the in vitro part, we achieved 81% of rooted plants in the IBA plus, metatopolin plus treatment, which is something we expected. For the second IBA treatment, we reached nearly 70% of rooted plants, which is, as I said, something we wanted to happen. However, something unexpected for us was this, that the ex vitro plants rooted as well, which is, as you should know, the callus in the plant absorbs nutrients from medium, and it happened to be that the callus absorbs nutrients from soil, more like a solution we were watering the plants. So the rooted percentage of ex vitro plants is pretty much the same in everything, but just 2 treatments showed like 56% of root. And now for some of the results. So fresh weight per plant in vitro in the IBA concentration is much higher since the roots, of course, because they weigh as well. In ex vitro, we can see the same thing, but not in the IBA treatment. We can see this in the metatopolin concentration, which indicates us that the leaves should be much bigger than in the ones that were not sprayed with metatopolin. For the number of roots, we can see as well that in the in vitro thing, we have a lot more roots than in the ex vitro, and the most amount of roots we achieved in the IBA plus conditions. And for the last thing, we will talk about total chlorophyll per fresh weight and the chlorophyll ratio in the plants. So total chlorophyll per fresh weight is pretty much the same in ex vitro, and we just happen to see only one, the metatopolin plus and IBA plus, which is really high in vitro. But the ratio of chlorophyll is pretty much higher in ex vitro parts, where the plants were treated with IBA, than in the in vitro part, when the same treatments, which, and I would want to say a thing about this, which means a higher chlorophyll ratio indicates a greater proportion of reaction centers relative to antenna complexes, reflecting a more effective photosynthetic system, typically associated with optimal growth conditions. A lower ratio suggests an increased investment in light harvesting complexes, which is often an adaptive response to suboptimal conditions such as low light or environmental stress. So the environmental stress is the part that was a main thing to why we use the metatopolin. As we can see, the IBA gives us good results in rooting, but gives us bad results in stress of the plant, which is still the protocol of rooting should be worked on. And as a end of my presentation, take-home message. So Quercus is a difficult species for in vitro propagation. Rooting efficiency is a major limiting factor, especially in mature material. IBA significantly improves rooting in both in vitro and ex vitro conditions. Optimization of auxin-based protocols is crucial for successful propagation. And as my body wanted to stop me, remember, even when the things don't stick to the plan, you can always finish and have a great success, as this ex-plant that fell down but had a spirit to fight for his life. So thank you for your attention. There's the QR code. If you want to contact me, you can feel free to scan. And that's all.
Thank you very much. And now please stay, because we have time for a few questions.
It's impressive work. I think for master thesis is really a lot. And even from this fluorescence of chlorophyll, you can make like a lot of information more than you show here. So you have a lot of data, and I think it will be very good master thesis.
As a lot of data, there is a special thanks to my supervisor, which is Mikołaj, of course. And special thanks from here for Dr. João Martins. That is, of course, pretty much a supervisor as well. From this point on, this was mainly his idea to do this experiment this way. We based on his protocols and his research, so pretty much the biggest thanks should be to him, not to me. Yes, there's a lot of master thesis. Should I be happy? Yes, of course, because the work will be great. Should I be happy about writing it? Not, because it's a lot to write about.
That's true.
So yeah, for an oak in vitro, there is a lot of things still to be worked on and discovered. This study that we did shows only this. There is much more to study on. This protocol is not great at all. It still needs a lot of improvements, and there are many more factors depending, like rooting of an oak. Even it can be the genotypes from the plants. We only work on one genotype in this master thesis, and imagine if you work on like 100 different trees. So yeah, it is a lot of work to do. Any more questions to me? Please no.
So I think that I don't see any more questions, and ladies and gentlemen, so I think that this session is just ended, and please welcome me just now starting the coffee break. It's time for coffee, I think.
And I will be leading this session on behalf of Tomek Leski, who unfortunately had to attend some personal matters. So without any further delay, let's begin our next session. And the first participant is Michael Brodsky, who will lecture a topic about putting a young forest under a microscope and seeing fungi. The floor is yours.
Yes, excellent. My name is Michael, as you said, and I come from Estonia. And I must say that if there are any serious mycology enthusiasts here, I might disappoint you, because the only species I'm going to name is Spice abies, the Norway spruce, a very widespread tree in Poland, as I gathered, and one of the most widespread easily in Estonia, one of the dominant species, makes up about a quarter of all the trees in Estonia. And yes, yesterday I learned that here in Poland the climate is predicted to become much more dry, and the precipitation levels are supposed to go down over the next century. And for Estonia and other northern countries, the predictions actually state the opposite. We are supposed to get quite a bit more rainfall. And this is what we are modeling in our free air humidity manipulation experimental area located somewhere in southeast of Estonia. We modeled the increase in precipitation. And for that, over there, we have some plots, 3 different kinds of plots. Of course, control plot where we just let the trees grow. We also have an air humidification plots where we add some about 3% of normal relative air humidity to the air and see how trees react. And the soil irrigation plot where all the moisture goes straight into soil, and that increase about is 15%. So, and it's mostly plant physiologists working there, and their main focus is, of course, the plant. And we see often that under the increase of air humidity, the trees grow a bit slower. And to compensate it, the deciduous trees, for example, tend to have somewhat longer vegetation period. They throw their leaves off later in fall. Right. And some of those our people had an idea to see how air humidity, increase in air humidity impacts the waxes on the surface of the needle. And for that they did some scanning electron microscopy. Resulting pictures look about like that. And yes, they investigated surface waxes from sampled needles that were about 1 year old. So they saw a lot of waxes. I'll talk a bit on that later. But they also saw a lot of fungi, and this is where I came in. I was given to these pictures to look at and to figure out if I can do something with them. And I could. So I asked a question for research. Could we maybe see from these pictures how increase in air humidity impacts the extent of colonization of fungi of the needle surface, and maybe we can even figure out if it can impact the colonization inside the needles. Right. And we had 2 different data sets. First one with a magnification of about 700 times, and we had 230 pictures of this. This is mostly focused on the interstomatal areas, right? These flat and smooth areas around the stomata, and a data set with 2800 times magnification that is focused on the epistomatal areas, right? on top of the stomata. Now, these words are rather similar, so stay alert, because I couldn't find any synonyms to make them more different from each other. So, of course, I didn't do everything by hand, because these days we have machine learning tools. And I took the machine and taught it to recognize these tiny little fungal hyphae on top of the needle. Right, the machine translated it to images that were initially black and white. I painted them orange to make them look nicer here. And then I stuck it into RisoVision, that is a common tool for anyone who deals with scanning roots or something like that, and that managed to turn the images into numbers with lots of raw data. And the raw data was initially in pixels, square pixels, and so on, so I had to do some measurements by hand to be able to translate those pixels into units more usable in real world, such as micrometers and millimeters. So I ended up with 3 data sets of, first of all, of mean hyphal network area in square micrometers per square millimeter. Second, mean hyphal length in micrometers per square millimeter. And the third one is the mean hyphal diameter in just micrometers. Now, these first 2, network area and hyphal length, they are strongly intercorrelated, so I will only talk about network areas, but the tendencies that we see, they also work with the hyphal length stuff. Now, the second thing, you may see these tiny little letters here. These mark the statistical groups, right? If these letters are different, if there are no shared letters, then there is a statistically significant difference between those 2 groups, right? And of course, we compare only top-down, not left to right. So, in some more details, with the data set regarding the interstomatal areas, we see the hyphal network areas. Now, on average, the hyphae cover about 1.5% of the needle surface we see. That's overall average of our whole data set. Now, you can see here that there is quite a significant difference, as in the treatment with increased air humidity, the average hyphal coverage is about 2 times lower than in control and irrigation treatment. Now, control and irrigation here are completely equal, as I think that the effects of irrigation, they stay underground, they don't make it upstairs, right? They don't make it to the aerial tissues of the tree, and only the humidification, air humidification, influences the fungal hyphae growing up there. So the difference here was quite pronounced. Now, we go to the other data set that was focused on the epistomatal areas, and here we can see there is completely no difference in this data. And the fungal hyphae here cover about 2.5% of the surface. Now, it's, you noticed, quite a bit higher than the previous one. It makes sense to me because it is quite difficult for fungi to grow on top of the needles or any other kind of leaves, because the weather conditions there are rather variable, unstable, and there are not a lot of nutrients there, so all the fungi try to get inside the leaf from what they gather, and the main way inside the leaf is through the stomata, as it provides a natural opening. Right. So our atmosphere treatment did not seemingly impact the fungi trying to get inside. Right. Now, there was also some difference in hyphal diameters between our different treatments, which was, of course, not quite as near pronounced as the difference in network areas, but it's not supposed to be. There's only so thin that the hyphae can get. Right. Now, the question, of course, is why could the air humidification be so different? One thing is that humidity takes out some spores from the air, and the other thing that I think was more important here was the waxes that other people looked at. So, unfortunately, this is better seen from up close and not very visible on the big screen, but under increased air humidity, the structure of epicuticular wax was different. In fact, it consisted very often of a lot of granules and tubules and tufts, while in control and irrigation treatments, the waxes tended to be smooth and very amorphous. That means they are more degraded as the waxes start out more like granules and tubules. Why is that? Probably it is some kind of a defensive reaction of the tree to increased air humidity, to increased stress that they suffer under. And it is probably also more effective at repelling the fungi from being able to grow on top of there. Right? So, as a small conclusion for the question, can we see whether the intensity of fungal colonization difference and in interstomatal areas, yes, easily we can, but we didn't get really any hints on whether the colonization might be more or less intense inside the needles, as we of course didn't see any difference and can't really see inside the needle for now at least. And so this time we concluded that effects of increased air humidity, they are probably confined to the outer surface of the needle. So far so good, and thank you.
Thank you, Michal, for your interesting presentation. Now please welcome our second participant, Tomasz Sobczak, who will present Hidden in Plain Sight, Artomyces microsporus in Polish microbiota. Okay, Tomek, floor is yours.
Hello, my name is Tomek Sobczak, and I'm going to present a topic that may be interesting to those that are interested in fungi. Because today I'm gonna talk about Artomyces microsporus and Artomyces, the genus, overall. The genus Artomyces consists of saprotrophic wood-inhabiting fungi that produce clavarioid fruiting bodies. They are often called coral fungi. And due to the shape of their fruiting bodies, this genus is called crown-tipped corals. Due to very difficult identification of morphological features in this group of coral fungi, members of this genus were placed in different genera like Clavaria or Clavicorona, and recent research in molecular and phylogenetic of Artomyces revealed about 15 species in this genus. They mostly differ in geographic distribution and host tree species. And here you can see two of the most widely known coral fungi, so Artomyces and Ramaria. And here you can see the crown tips, so they are like ball or crown shaped, as opposed to Ramaria, which has straight or blunt top peaks or apices of the fruiting bodies. So in Europe, there are currently 4 species of Artomyces. Artomyces pyxidatus is the most widely known. It grows in riparian broadleaf forests. It is red listed in Poland due to declining ranges of riparian forest. It has a global distribution. Artomyces microsporus was described in East Asia in 1992, and in 2004 it was first reported on pine trees in Ukraine, near Kiev, and it was reported from some more sites since. Artomyces cristatus is very rare and is known only from Fennoscandia, mostly some records from Russia also, and it grows in old growth conifer forests. It is also present in the USA, and Artomyces piperatus is only known from one single stand in Finland. As you can see, they present rather similar morphology. The main differences are, like I said, the host trees and the distribution. In Poland, Artomyces pyxidatus is widely distributed, but as I said, its range is declining due to drying of riparian forests and subsequent processes that follow this. And during our research trips to Pomerania region, we were researching Auroboletus projectellus. We have found fruiting bodies of Artomyces microsporus growing on pine trees. And at that time, that was the westernmost occurrence of this fungus. And it was growing on pine tree, which is the most important tree in a Polish forestry. So we thought that it was only reported recently in Europe, so we thought that it was maybe invasive, maybe foreign. And we wanted to evaluate how much it could be spread in Poland. So we also thought that the morphology is very similar between the species of this genus. So we thought that maybe somebody found it previously, but just didn't know that the species existed. That happens. So we reached out to various fungaria, to professional mycologists, and also amateurs on like Facebook groups and iNaturalist to send us samples of Artomyces fruiting bodies. We have examined these specimens using combined molecular and microscopic analysis because some of these specimens were really old, and it's very hard to produce intact DNA from old samples. And we also wanted to evaluate the potential distribution of the species using Maxent modeling. So here are some findings from our specimens. The micromorphology of Artomyces microsporus is well described at this point, and we have found one feature described in 2003 by Professor Edward Leakey. It is two sterigmate basidia that occurs in microsporus but not in pyxidatus. The picture is maybe not that clear, but basidia are usually very hard to see. And one feature that was described by Professor Leakey is that this microsporus supposedly grown on hardwoods. But I reached to Professor Leakey, and he responded that the identification of the substrate was carried out using a microscope based on the debris found in the packet with holotype. And after those 23 years, he concluded that it was probably a misidentification, and it was probably growing on Pinus densiflora in Japan. Okay, and here are also more microscopic features. The name Microscoporus implies the smaller size of the spores, and it is very visible under a microscope. The most reliable feature is the Q values, average Q values, so the width-length ratio of the spores. And as you can see, Artomyces microsporus has about 1.4, and Artomyces pyxidatus about 1.7, and it is consistent in our collection and also in the literature. And also we have compared our specimens, ITS sequences to UNITE and GeneBank databases, and here you can see the similarities between our collection and the collection from Ukraine. You can see that the percent of query coverage is 99.27, so it's almost a perfect match. And you can see also the comparison to the holotype from Japan. So the coverage is a bit lower, which indicates that our samples are more closely related to the Ukrainian collection, and there's also some difference between the geographically separated population of this species. This here also is from China, and we can see that the difference is a bit bigger. And okay, so overall we have collected 74 specimens of Artomyces from various collections. On the map on the right, you can see the collections, the geographical distributions of the collections. The circled locations are our study sites of Aureoboletus projectellus, Kampinoski National Park, and Bieszczady Mountains. Those are some bigger projects that were carried out, and they provided a bulk of the records. And using our combined approach, we have found 26 collections of Artomyces microsporus that were identified as pyxidatus, and the oldest data was dating back to 1965. So over 20 years before the formal description of the species, and almost 40 years before the description of the species in Europe. And also we have confirmed that the Artomyces microsporus was only found on pines, two-needle pines. In Poland, it was almost always Pinus sylvestris and one Pinus nigra. And Artomyces pyxidatus was only found on deciduous trees, mostly Populus and Fagus, but also Fraxinus. It is sometimes hard to identify the host tree because they grow on severely decayed logs. And the results of microscopic and molecular analysis help us to confirm the macromorphological differences, because there are also some differences in macromorphology. And like extrapolating from our findings from other analyses, we have confirmed the distinction between these two species. Okay, and to further research for the maxent modeling, we also included records based on photographic evidence, because we can like identify them from the looks alone. And here are some comparisons. They look very similar, but Artomyces microsporus is much more densely packed, and the crowns on the top have 10 plus branches growing from each one. And Artomyces pyxidatus has almost always below 10 branches and is more loose in the overall habit. And it grows on various hosts, but mostly like broadleaf trees. And as I said before, the microsporus grows only on pines. And here is the map of all the records that we have identified. As you can see, here on the right, there are the original collection in Japan, 3 in Korea, some in Russia, and most of these are in Europe. But due to scarce population in these areas between Japan and Europe, we excluded these records from the modeling because the distribution in these parts is probably much wider, but it is just poorly documented, and it would probably result in artifacts. And in the final data set used for modeling the distribution of the species in Europe and in Poland, we have obtained 130 records. We cut off the range of the records on the Ural Mountains, and we also reduced the data set to 1 record per 2 kilometer radius to avoid biases, because that would make the site that more records were made appear as more suitable, and that's not the fact. And first we used we determined the 5 most important climatic values, and then we have added also 3 soil variables, soil type, soil carbon, and soil pH. And the most important variables climatic variables determined to be mean temperature of the coldest quarter, mean diurnal air range, mean temperature of the wettest quarter, mean temperature of the driest quarter, and also precipitation of warmest quarter, as well as soil type. And here you can see the distribution of the predicted distribution of Artomyces microsporus. And on the right, you can see the native range of Pinus sylvestris. And the occurrence in continental Europe is closely correlated to the distribution of Pinus sylvestris. But as you can see, the distribution in Phenoscandia is very limited. And from our models, we have found that this is most likely to the soil composition. Because before the inclusion of soil type, more area in Scandinavia was marked as suitable for the species. Okay, so to conclude, our findings suggest that Artomyces microsporus is a widespread species in Poland, in all of Central Europe, because previously it was thought to be rare or recent introduction. We have confirmed 2 needle pines as exclusive hosts for Artomyces microsporus, Pinus sylvestris, Pinus nigra, and Pinus densiflora in Asia. And our findings suggest native rather than introduced status of this species. And also, as I said before, the distribution strongly correlates with distribution of Pinus sylvestris. And also another addition, that examining the herbaria specimens of fungi is just a goldmine of data that we are just sitting on, and we are not really paying attention to. We can probably get maybe dozens of or hundreds of new species for Europe just by now analyzing what we have in herbaria. Okay, and that's all. Thank you for your attention.
Thank you for being on time. So let's move on to the next topic, genetic diversity and ecological niche assessment of silver fir conservation resources in the Sudets, which will be presented by Jan Sus from Poznań University of Life Sciences.
Okay, so hi everyone. My name is Jan Sus. I'm a PhD student at the Poznań University of Life Sciences. And today I would like to present you a study evaluating genetic and ecological diversity of clonal conservation seed orchards established as a part of the silver fir restoration program in the Sudetes. So a quick introduction to the program itself. The program was established like about 30 years ago because of this small share of the silver fir in the Sudetes at the end of the 20th century. It was about 0.4%. Main causes were, in fact, like the human impact, in fact, growing human pressure since Middle Ages, and then later forest management practices has contributed to this process. And there was a problem because of insufficient seed base for reforestation actions. And this gave rise to this concept, to this idea of developing such a seed base by establishing from remaining individuals conservation clonal seed orchards and clone archives like intended to both preserve the genetic pool and also provide the reproductive material for reforestation. And here is some big mistake I made, and I've noticed it during the coffee break. It shouldn't be northwest and northeast, but southwest and southeast. But basically, the program was based on local resources, and it was partially because the genetic distinctiveness of Sudetic and Carpathian silver fir, but also because of this belief in local adaptation and that the local is the best. And there was like the selection, two selection criteria, and they included vitality traits, so basically crown vigorous enough to allow sampling, and also local origin inferred from the diameter at breast height, just as the indicator of long-term radial growth. And the program's target is about 80% of coverage area of the species in the Sudetes, and currently we have about 2.5%, which represents a significant achievement, but although it is still far from this intended goal. And here is something else, like I said about the seed base, and this is what the seed base of silver fir at that time looked like. And it was about 120 hectares of seed stands, in which silver fir share was often about 20 to 30%. And we also had this 11 individuals, like individuals selected as a reliable source of genetic material. And also here you can see like the cone production from a stand established in the Sudetes in the 1964, and you can see that the cone bearing starts relatively late. It's about at around 30 years of age, and after that it is quite irregular. So this is like another problem of silver fir regeneration, like it was at that time. And based on this knowledge, we have defined like 5 key goals. So the first one, determining the level of natural genetic diversity and its distribution among clonal conservation seed orchards, to see if there are some seed orchards that have like very high or very low genetic diversity, characterizing genetic diversity in relation to natural silver fir population in Europe, to compare some basic genetic parameters with natural population silver fir like across the European range of the species. Identifying the spatial genetic structure of the species to check if there are some differences between different parts of the Sudetes, predicting the dynamics of the identified cluster under a mild climate change scenario to check if there are some patterns in like shifts of the genetic boundaries between those clusters, and also comparing the ecological niche preserved in orchards with the current ecological niche occupied by the silver fir to investigate this like ecological representativeness of seed orchards. So we have here the map with the locations of conservation trees, like these maternal trees from which clonal conservation seed orchards were established. And these are mapped at the level of forest compartments. We analyzed a total of 8 clonal conservation seed orchards and 3 clone archives. And I will refer to those from now just as clonal conservation seed orchards, to both of these like objects, to simplify things. And from these 11 seed orchards, we studied the 1,258 individuals using 12 nuclear microsatellite markers. So as I said, we investigated the neutral part of, like neutral component of genetic diversity. And here are some basic information of the clonal conservation seed orchards and these 3 clone archives that I've mentioned. They are located within the Karkonosze National Park, so somewhere here. And they are highlighted in green, so these are the 3 at the bottom of the table. And because the initial grouping in seed orchards followed an old seed regionalization system, we divided them, the individuals, into 18 ecogeographic groups, which we believe provide like a more natural clustering of these individuals, because as you could see, some of the like maternal trees within the seed orchards were highly dispersed across the Sudetes. And for example, these 3 groups these are in fact one seed orchard. And so here is the table with some basic genetic parameters, and we investigated 12 papers. In general, it was 184 populations across the European range of the species, and as expected, genetic diversity was slightly lower than in natural population, but there was like pretty high values of mean number of alleles and also allelic richness, which suggests that a large amount of genetic variants have been like preserved, maintained in those seed orchards, with about 68% of natural population having lower or equal values. And with the heterozygosity values, they were like, you can say, modest, which can point to some turbulent past of the Sudetic fear, and also provide some evidence, some trails of like inbreeding within the parental trees, with only about 35% of natural population having lower or equal values. And here is the genetic clustering analysis based on those 18 ecogeographical groups, and it shows like this spatial differentiation between the eastern and western regions. So we have like this yellow or slightly orange color, first like cluster, and in the western region we here dominates the dark blue maybe color. But you can also see that within the western group there there was like identified 3rd cluster, so the light blue, and this these are the 3 clone archives located in the Karkonosze National Park, so you can see it here. But conversely, we also observed like this considerable mixing of clusters. This points to rather efficient gene flow between, like across the entire Sudetes area. And here also is the table with seed orchards ranked by their like genetic contribution to the overall genetic pool, like Sudetic gene pool. And for example, you can see that Yugov is on the top, and because like it contributes, its contribution is the most like contribution of alleles, because it generally has the most alleles of all the seed orchards. And then Maciejowa's contribution is just 21 like unique alleles to the gene pool, but we also have this last column with private alleles, so genetic variants that are unique and that are present only in specific seed orchards. And as you can see, there is like rather uneven distribution of alleles among the seed orchards, and this is potential risk of losing certain genetic variants if any of this seed orchard will be lost in the future for any reason. And here's like the projected changes based on this genetic clustering and also bioclimatic variables. And this selected scenario, it assumes, as I said, mild climatic change consistent with the EU emission targets aimed at keeping global warming below 2 degrees Celsius, in relation to the pre-industrial era. As you can see, there's like this dissolution of these genetic boundaries for most of the clusters, but we have this one cluster which shows significant
expansion, and this expansion involves individuals from the Kachavsky Mountains and potentially reflecting some unique environment and condition of that area. But as the study focuses solely on natural genetic diversity, this pattern that we observe here should be validated by analyzing the adaptive component of genetic diversity, adaptive potential of individuals across the Sudetes. And here's the niche comparison between the environmental space occupied by the mother trees of colonial conservation seed orchards and current niche of silver fir. And the latter was defined based on all forest compartments containing silver fir sourced from the forest data bank. And like this green area is stability, so just like the overlap between both niches, the red one is expansion, so basically space occupied by the mother trees that is currently not inhabited by the silver fir, and unfilling is the opposite, so it's the niche currently occupied by the silver fir that is not represented by the mother trees. And we also calculated two overlap indices, Schoner's D and Hellinger's I, and they were pretty high. We also conducted the similarity test to check if there were some biases created during the sampling, and because of the lack of statistical significance, we can say that this overlap that we see is driven by, let's say, regional environmental availability rather than some specific sampling, like selective sampling strategy used. And to sum everything up, we can say that the established seed orchard successfully captured the genetic diversity comparable to the that of natural population, and there's this one problem with uneven distribution of genetic variants. Then the subtle genetic differences between western and eastern parts of the Sudetes, this might be relevant for just seed transfer like from west to east and east to west. CCSO, so colonial conservation seed orchards, show high representativeness, about 18%. We also have this 20% niche expansion, which might be potential buffer because these are the mother trees that was like living in the climatic conditions that are not currently occupied by the silver fir, and also this unbiased environmental reflection that we detected. And even under this mild climatic change scenario, most of the identified genetic cluster, they like lose the importance, decline in importance, which may have some implication for long-term persistence of the stands. But as I said, we should focus now on the adaptive potential, adaptive part of the genetic diversity. And this would help to confirm or not some genetic patterns that we see here, and which help or we justify or not justify more localized approach for the management of genetic resources in the Sudetes, and of course continuous monitoring of established stand is essential to assess their like potential, adaptive potential of adaptive potential under changing climatic conditions, and also to support selection of the most appropriate strategies to mitigate the negative impacts of these changes. And thank you for listening, and I would like to thank the State Forest National Forest Holding for their support.
Thank you, Jan, for your interesting presentation. This means that we actually crossed the halfway point of our session. And the next participant is Martyna Lasek, who will present a topic, Balancing Genetic Integrity and Adaptive Potential Under Climate Change, the Scots Pine Case. Martyna represents Institute of Dendrology, Polish Academy of Sciences.
Thank you, Hania. Hi, everyone. My name is Martyna, and as you heard, I'm a PhD student in the Institute of Dendrology, and today I want to show you a small part of my PhD thesis, which is actually now under review, so these are all unpublished data yet. Okay, so There is no need to introduce Scots pine to any of you. However, it's worth mentioning that it's a pioneer species of wide Eurasian range. And due to the vast variety of inhabitat stands, it developed nearly 160 phenotypical variants called ecotypes. And because of that, it's highly economically valued species and generally major forest-forming species in Poland, providing 60% of total timber production. So in response to the wide variation in phenotypic traits and diversity in stand performance of specific origins, forest managers introduced the concept of regions of origins to protect the sources of forest reproductive material of exceptional value. And in Polish forestry, two classification systems are now of most particular importance. The first one is natural forest regionalization, and the other is seed regionalization. Both of them are used to delimitate regions of provenance for forest reproductive material. And you can see it right here on this picture. It's for each managed species in each European countries. And in Poland right now, we have 24 such zones just for Scots pine, and it's in effect since 2015. And Scots pine, just like many other species, is under strong pressure of progressing climate change, high temperatures during summer months, prolonging droughts, rapid rains, floods. This all leads to increased mortality and may cause changes in the distribution of this species. So the main aim of our study was to examine whether differences in productivity among population representing Polish provenance regions are more strongly associated with local climatic conditions than with genetic differentiation, and what this may imply for seed sourcing under climate change. And to check it, we used 27 populations of old-growth forest from Poland, and they all covered 24 seed zones, all of the provenance regions that you saw before. We sampled phenotypic data such as height and diameter at breast height for over 800 trees. We also performed genetic analysis based on PIC 50K SNP array. I hope I pronounced it correctly. So these were genetic analyses just for a background, and also we acquired climatic data for each of those populations. And hypothesis is that within the relatively narrow climatic gradient of Poland, differences in productivity and phenotypic differentiation among spotted pine population across are associated mainly with local climatic variation rather than neutral genetic structure. So this is first of our results, and it's a box plot showing variation of diameters and heights of pine trees among examined stands, with a division into two forest types, conifer and mixed. And as you can see here, the variation is huge, and in terms of height, there is even a 12-meter variation. And it must be said that these are not regular stands. They are all classified as superior and just perfect for seed sourcing. So even within it, the differentiation is huge. But for a more clear visualization, we performed a spatial comparison of phenotypic and genetic traits across the studied populations. And as you can see, the DBH and H, which are diameter, breast height and height, they doesn't form specific patterns. However, as you can see on the C graph, which is FST value, a value basic for genetical differentiation, we can see some kind of pattern, like the lowest FST is here, and it goes more diverse northwards, northeast side. And I can say that it might be corresponding with the side index, because what you can see at the D is basically, okay, I thought that maybe we doesn't see a pattern differentiation in just age and just DBH, but what if we mix it together? So I calculated the base, the side index for the base age of 100 years old, because all of them were much, much more older, even 220 years old. And this is what you can see here. And there is a pure correspondence for me that those who are better performing the northeastern side also has a higher genetic differentiation on a neutral ground. That's what, there are not outliers, it's just a neutral genetic background. And looking at the climatic variation, I really like this analysis. Here you can see where Poland sits within the European climatic range. So the green dots is Polish climate right now. The gray background is European stance, Central European stance, and the lighted gray is the rest of European stance. And these three colors is just Polish climate under two climatic future models, RCP 4.5 and 8.5, and the current one. So even now, a Polish population are rather in a dry and warm climate, so it's a nice point to continue. So we did the side index prediction, and we performed climate projections to assess how the relative productivity of this population may change by the end of the century. Using linear models, and we found that there are three main groups of climatic correlates: thermal conditions, summer moisture deficit, and early spring conditions. It's here. And this is very interesting for me because those populations who were before superior and best of the best, like Bolewice, for example, here, or in the North Poland, Miłobłyn, will be declining, almost dying off and underperforming, while those now worst, like Włocławek in this middle Poland climb, are going to be stable or even improving. So there are losers and winners in here. And for going to the conclusions, variation in productivity among the analyzed Scots pine populations in Poland was more closely associated with climatic conditions at the onset of growing season than with the summer conditions alone. The high support for the spring Pacific model suggests the early season temperature and moisture conditions are important correlates of site productivity across the studied stands. The future models suggest that the most productive population will experience the greatest declines, while currently underperforming are expected to remain stable. The observed differentiation was not reflected by neutral genetic structure alone, and it's likely shaped by the combined effects of local climate, stand condition, and potentially adaptive variation. And because of occupying transitional climatic zone within the species European range, they may represent valuable material for future testing in breeding and assistant migration programs. And also this result supports a cautious re-evaluation of seed transfer strategies and highlight the need to combine phenotypic, climatic, and genomic evidence when identifying Scots pine population best suited for the future environmental conditions. Thank you for your time and attention. Here is my research page if someone would like to read more about that.
Thank you, Martina. You saved our time. Thank you very much. And our last but not least participant is Usam Samudi from National Research Institute for Agriculture, Food and Environment and University of Bordeaux. And she will present genomic maladaptation to climate change with population and adaptive potential of Scots pine. The floor is yours.
Thank you for the introduction. Can I take the mic or not? Thank you. Hello, everyone. I hope you still have a little energy for the last presentation of the morning. So I'm Mausem Samouji from the National Institute of Agriculture and Environment in Bordeaux in France. And today I will talk about my master internship project, which is about genomic maladaptation to climate change within populations and adaptive potential of Scots pine. So this project is supervised by Thomas Francisco and Santiago González Martinez. So forests are vital to our ecological balance in society, holding up 80% of the biological terrestrial biodiversity. And it is without to know that trees with long generation time and low dispersal abilities are highly vulnerable to climate change with rising temperatures and extreme weather events. So here are some pictures that are illustrating some drought in forests that shows that there is a potential vulnerability of tree species to climate change. Then the question is, what is the effects of climate change on tree species? And one of those is maladaptation. Well, maladaptation occurs when phenotypes of individuals are distant from the optimal phenotype in a given environment. So here I will use a metaphor from Brady to illustrate this phenomenon. Well, the target is illustrating the fitness landscape and the arrows individuals of a population. So we can see here that the arrows that were before at the center of the targets were not, well, is not adapted anymore to the new targets in a context of climate change because the environment is moving. It's what we call the moving target. So this is the maladaptation concept. And the rapid shift in environmental condition is one growing cause of maladaptation. So recent developments in genomics allowed us to assess this maladaptation, and one of them is the genomic offset. So the genomic offset corresponds to the magnitude of genetic shift. In our study, it will be in allele frequencies shifts required by a population to maintain a current fitness under different climates. So here the graph helps us to understand what we do when we apply this approach. So first, we are establishing a relationship between the genomic composition and the climatic gradients. So here it is representing by a linear relationship. And then here we have the current genomic composition of an individual or a population. And if we suppose that this population or individual undergoes climate change for the same genomic composition here, we will probably have a gap between this point and the future required genomic composition. So this gap is called genomic offset. Then in our study, we will focus on Scots pine. Well, I think you already know something about this species. So here we have the large species range across Europe and Asia of Scots pine. So the fact that this range is particularly large make this species particularly at risk with climate change, especially in the south of the range. And it's also covering different environments, so it was a good model for us. Then in red we have our sampled population that we obtained thanks to different projects and programs. So it's pretty well covering the range, especially in the west of Europe. Then we want to answer to a general question, that is, what impact does climate change will have on Scots pine and populations, and what is their adaptive response potential? So first, as I said, to establish this relationship between the genomic composition and the climate, we used the Genotype Environment Association method, so the GEA. So we use this to identify climate-associated losses. So first, at the beginning, we had around 35,000 SNPs from our 79 populations, covering our range. And then we also selected 7 bioclimatics variables. And after that, we applied our 6GA methods. So linear ones, non-linear ones, corrected or not by population structure. And combining those methods helped us to reduce false positive and to reduce biases due to our initial assumption. Then, thanks to all methods and at the more conservative threshold, we obtained 1,800 candidate SNPs for our model. So we imputed them in an RDI model, a redundancy analysis model, in order to perform our genomic offset. So after this, we projected our results on a map, like here, that represent the spatial representation of genomic offset between the current and the future climatic condition across the range. So more particularly, it is the prediction for the 2041 and 2070 period. And it is basically the average of 5 climate models for the SSP3 and 7 scenario. Sorry. So here I have to explain that the highest value corresponds to the population that has the highest degree of maladaptation. So here we can see that on the Riviera coast here, so in the south of France and northern Italy, we have the population most maladapted, with the highest value of genomic offset. Also here in Spain and a little bit at the west of Scotland. So to conclude for the first point, we obtained climate-associated SNP thanks to our methods, and we built a predictive model of genomic offset across the species range. And we observed that in the south of France, north of Italy, and west of Scotland appeared like population particularly maladapted to future climates, according to our models. But our study still have some limits. So for example, for the moment, we have no link between the genomic offset and the prediction and the fitness, which is really relevant to see if our models can be validated or not. And we have only prediction at the population level and not the tree level, and no information on adaptive potential. But I don't know if you remember the graph of genomic offset, but we were supposing that the genomic composition does not change to apply this model, so it can be really important. That's why for the forthcoming steps, the current one, because we are doing the analysis, we want to validate our genomic offset model with phenotypic data obtained on common gardens, and also investigate the patterns of climate maladaptation within 2 populations across cohorts to gain insight into the within population variability and also the adaptive potential of those populations. So in the second point, as I said, we will focus on 2 populations. So one is located in Finland and the other in Scotland. And for each population, we have 500 genotyped adults and 250 genotyped juvenile. And what we will do, basically, I will reuse the metaphor from Brady. So first, we will want to assess the individual climate maladaptation variation. So basically what we will do here, it's measuring the distance between the genotype optimum and the actual position of the adult tree. So this is called the adult climatic maladaptation. So we will do this for all individuals and see if there is some variation between them. And after this, we will see if there is a rapid genetic adaptation happening within population in response to climate change. And basically, we will do the same. So for the adult trees and the pink ones are the juvenile trees. And so we will just compare the average of juvenile climatic maladaptation and adult ones. And if the juvenile ones is lower than the adult ones, it could mean that there is a rapid genetic adaptation just across one generation, and that could mean that there is maybe an adaptive potential in those populations. So as I said, we are currently doing this analysis, so if you are interested to talk later about, I would be happy to answer to your question. Thank you, everyone.
Thank you, Mayusa. So we have 30 minutes for discussion, so let's begin the Q&A session. Who has the questions for Mausam? Mausam, yeah. No questions? Are you sure? Okay, so for the purpose of the stream, I will ask our first participant, Mihail, to the podium. If anybody has questions for him. Is Mihail in the room? No. Okay. So, questions to Tomasz Sobczak? I will give you another second. Okay. So the next participant was Jan Sos. Any questions?
So thank you for your presentation. I was wondering if you're not afraid of, because I saw on your structure analysis that you didn't have even number of individuals within population. So don't you think that this might somehow bias your results?
By the, you mean genetic clustering or just parameters?
Just
of
clustering.
Like genetic?
Yeah, just clusters of genetic differentiation.
Well, it might be a problem here, but in fact we somehow manipulated this data by grouping them in this ecogeographical group. So this itself like results in some different structure of like genetic structure, and the problem is that we have as much individuals like clones from the seed orchard as there is, because some of them there was more of them, but not every clone was able to survive. So we just took what was left and we were working with this. So.
Okay, thank you so much.
Any other questions to Jan?
Thank you for the presentation. If I understood it correctly, the aim of the Polish side of the cities is to increase the population of silver fir or the representation of the silver fir. I wanted to ask you if whether you know what is the most common way to introduce or rather reintroduce the silver fir into the forests, like if you use seedlings, if you use saplings, or how do you interpret the silver fir into the forests?
Okay, so asking me about like the silviculture methods of introducing.
Yeah.
Well, I'm not sure which one is the best because I didn't like investigate it, but of course the best way, like the most successful way, I suppose it will be to use some mycorrhizal seedling or like enhance the seedlings within like the forestry like school, and probably that, but the problem is maybe with the main aim of the program was basically to preserve the genetic pool, and that was it, but also to preserve it for future reforestation measures, but it's not yet like clearly maybe it's not clear what is the best way, at least for me, for now.
So right now you are trying to stabilize the populations, but it's for the further research how to maintain the population. Do I
Well,
understand
I can say that the main aim of the study was to, in fact, check if the strategy that was established, like that was used for the conservation of the gene pool, was successful or not, because it wasn't investigated. Like it was based like on preserving genetic pool, but without genetic data. So. Yeah, basically, yes.
Okay, thank you.
Thanks.
Okay, are we done here, or are there any other questions? Nope. Thank you, Jan. So let's move on to Martyna. Okay, I can see that we can close our Q&A session. Thank you all for the participation in our midday session. I think that we can go to a break, and in 25 minutes there's going to be the lunch. So thank you all once again.
and without further time losing, I'm inviting the first speaker, Miss Aurora Bonando from Tuscany University in Italy, who will present an interesting application of GIS system. Thank you. Okay. An interesting application of spatial analysis for the selection of the harvesting system. I repeat, you know that each one of the presenters has exactly 15 minutes, and the questions will be at the end.
Okay. Hello, everyone. I'm Aurora, and as Francesco anticipated, I will talk about a decision support system for the forest operation planning. But let's start with a little bit of context about the small-scale forestry in Italy. Especially in the central-south Italy, logging systems are often chosen at the project stage by the local forester or even left entirely to the contractors carry out the forest operations. Obviously, this leads to low transparency in decision-making and lack of standardized evaluation criteria. Addressing this lack is particularly important in environmentally sensitive areas where forest management must support the multifunctionality of forests. So we start from this need for an objective and transparent procedure for the selection of logging system. And we saw in previously studies that the GIS HP is a promising procedure for this purpose. However, forest operation planning is complex because involves different stakeholders with different roles and different perspectives. So our study aimed on the one hand to validate the GIS HP decision support system in unexplored forest type, and also is very typical in the central Italy, the chestnut coppices. And on the other hand, to test how robust are the recommendations when different stakeholders groups provide the expert judgments. The study was conducted in the municipal forest of Rocca di Papa in the Castelli Romani district, a beautiful district far only 25 kilometers from Rome. And I'm not saying that it's beautiful because I'm from Castelli Romani, but because, as you can see in the 3D images, in this area we can find woods and 2 lakes, because this area is part of an ancient volcano called Vulcano Laziale. And for this reason, here we found these fine textured soils and hilly landscapes. And the dominant stand structure, as you can see in the images in the right, is the chestnut coppices with standards. And the actual forest management plan divided the area in 233 forest management units. But for this study, we analyzed only 162 units. We decided to focus only in the actively managed one, excluding the not productive. Here, the logging system that were assessed. On the left, the extraction system, and on the right, the wood system. The difference is that the extraction system refers to how the material is, to what machinery is used to move the material out to of the forest. And the wood system instead describes how trees are processed, in which form the wood is extracted. And the 3 extraction systems that were considered for this study is a cable skidder for wood and all-terrain cable yarder. And the wood system is the cut-to-length, in which the trees are delimed and bucket in the forest near the stump, and the short logs are extracted. The tree-length system, in which trees are only delimed and long stem are extracted without the crown, and the whole tree harvesting, in which the whole tree is extracted, including branches and crowns. The methodology for the development of this decision support system starts with the data collection of 6 criteria. These 6 criteria represent the main constraints affecting the feasibility of harvesting and wood system selection. These criteria include data about the terrain condition like slope, roughness, and soil bearing capacity, data about accessibility, like root density and extraction distance, and also data about the stand, like the extraction timber amount, that it's also relevant to the operations. On the right, you can see how each criterion was collected, and then they were implemented as an input layer in our GIS database. For the GIS AHP methodology, we follow the procedure proposed by Latterini et al. In 2022 that can be summarized in 3 main steps. The first is the creation of one raster for each criterion and for each extraction system. And then we reclassify this raster into 5 suitability classes. So at the end of this first step, we obtain 18 reclassified rasters. In the second step, we apply the AHP methodology. We ask to 4 different stakeholder groups, researchers, technicians, owners, and forest workers, to rate how important from 1 to 9, that is the static scale, each criterion is important for the selection of the extraction system. The same was asked for the wood system, but they asked to rate the importance. And then these scores were used to compute the final stakeholder specific weight. In the last step, we return back in GIS when we apply a weighted average between the 6 criteria and their AHP weight. In this way, we obtain 4 maps, one for each stakeholder category. These maps identify for each forest management units which extraction system is the most suitable. And the same logic is then applied to the wood system in order to obtain other 4 wood system recommendation maps. Here the result about the extraction system. You can see the pattern is very clear. The cable skidder, the green one, dominates across all the area and across all the stakeholders group, with a full agreement in about 92.5% of the area, and with a minimum of recommendation of 94% in the categories of owners, and with a maximum recommendation of 100% in the categories of technicians. The disagreement concerns only 20 forest management units, and in the most of these cases, owners suggest the forwarder, while the other categories continue to recommend the cable skidder. And as we'll discuss later, this shifting is influenced, of course, by the stakeholder preferences, but also by a decision rule that we introduced to assign the final system. When the analysis is translated to the wood system, the same overall logic is still visible, since the skidding extraction is the predominant. The recommended wood system is mainly the wool tree harvesting, and here the full agreement is even higher, 96% of the area. And the disagreement concerns only 8 forest management units, when one of the stakeholder groups suggests the tree-length system instead of the wool tree harvesting, and the cut length is never selected as the best option in any of the units. So why the differences were so limited? The limited divergence can be explained by the fact that the stakeholder preferences were strongly shaped by the strong site constraints. In particular, the very low soil bearing capacity of the zone due to the volcanic soil really limit the high mechanized ground-based solution such as the forwarder. At the same time, the aero density of this area penalized the cable yarder. That is a solution more complex and more costly. So generally, it becomes more competitive when the ground-based solutions are not feasible. And despite this reason, most of the observed difference in the recommendation are due to this time-breaking rule that we introduced to avoid leaving any forest management units without an assigned system when the suitability scores are equal. And in these cases, we choose to assign manually not the forwarder as the extraction system, because it's generally the one with the highest operational efficiency. So these units with the equal score should not be interpreted as a true stakeholder disagreement or as a real superiority of the one system. In conclusion, the JSHP approach proved to be a useful and valid tool for bringing the objectivity in the identification of the suitable logging system, and proved also applicable in the central Italian chestnut coppices, and also robust because of the high level of convergence across the stakeholder groups. But I really want to highlight that the value of this method is not to remove the expert judgment, but to make it explicit and open to discuss, which is very important for improve the transparency in forest operation planning, especially in context like Italy, where logging system choices are still often postponed to the executive phase. Thank you for your attention.
Thank you, Aurora. Now let's stay in Italy, a bit more northern, and shifting the species from oak to poplar. And I invite Ecology Di Marzio, that always remain in the domain of harvesting.
Okay. Good morning, everyone. My name is Niccolò Di Marzio. I'm from Padua University. Today, I'm very happy to be here, and I would like to present a study that we concluded a few months ago, which is focused on a silvo-arable system. But before going into detail of this case study, I would like to just briefly introduce my PhD project, so that you have a framework to better understand the application of this study. The project was developed within a Forestry 4.0 transition that follows the Industry 4.0 transition, and it's basically focused on high-resolution data for forest operation planning. What we are doing is basically exploring technologies based on digital data and high-resolution data to better understand how machine traffic interacts with terrain, with infrastructure, and with soil vulnerability. These are some examples of interaction, not between me and the soil, but between machines and soil. You can see on one side an example of heavy interaction. I was basically inside a machine trail that was about 70 centimeter deep. And in the other case, I was still in a site where forest harvesting took places a few years before, but it was a case where the recovery was in a good state, and sometimes the machine trails were barely visible. My project was developed, as Francesco said, in the north of Italy, between the Veneto and the Trentino region, in the alpine area, alpine context. And in this area, the management response to the increasing severity and frequency of disturbance events, which is a European or even a global trend, is now the primary challenge, especially in those areas where the good quality wood production is a priority. This picture is from 2018, from the Vaja storm. I think that 1 year before you had something similar in Poland, in 2017. Nowadays they are still facing problems for the outbreaks of bark beetle, of course in the remaining stands. So in a first study, we took an overview at a very broad scale, and we wanted to analyze the relationships within the salvage logging areas of the machine and accessibility, trail density. This pink line that you can see are actually the machine trails that we digitized one by one. We had at the end approximately 800 kilometers of machine trails, the terrain constraints, and the hydrological sensitivity. Those red and blue areas are depth water areas. That is, in a nutshell, is areas where you can find permanent or temporary wet soil compared to the rest of the parcel. So we can define them sometimes as also sensitive sites. If in this case we try to understand how much and where machines were moving at the very broad scale, then we wanted to go a bit more into detail of the operational scale, and this is the study that I'm going to present today. We moved to an agroforestry site, and we made an assessment of the productivity and the soil response under real working conditions. We made the study in an experimental site where we had poplar rows between two agricultural cultivations, and this pattern was in the whole area. Here you can see the 4 poplar rows that we selected for the study and for the survey. Once again, we are in the north of Italy, but not in the Alpine region, but we are in the bottom part of Veneto region, which is called Rovigo. This is an example of agroforestry and alley cropping sites that we analyzed. Here you can find, for example, corns or any other grain cultivation. And these are the poplar rows that during the survey they had to be cutted. This is the main part. Within the operations, we selected, first of all, 2 different systems that were working in the field. One system was a semi-mechanized system composed by operator with chainsaw and an excavator that was helping during the operations, like, for example, holding the tree and moving and extracting the material to the roadside, and a fully mechanized system with forest harvesting. This is basically a machine that it's mostly used in the forest, but in this case they were trying this machine in this agroforestry system, and we basically compared system performance during the cutting and processing operations and the effect on soil that was produced by those machines. As I said before, we selected 4 poplar rows between the whole area, and of course some control areas where the operation were not carried out. We made, first of all, a time motion study to measure and assess the felling productivity and the processing productivity. Then we collected a lot of soil samples between the rows in different transects. We brought those samples to the laboratory, and we calculated the bulk density, the moisture content, the organic matter, and many other parameters. And we measured the penetration resistance, so the resistance of the soil to the penetration of this instrument from 0 up to down to 80 centimeters, or more. And then we used the drone to have drone flights and get orthophotos, and we used those photos for the calculation of the trafficked area. Going to the results about the performance evaluation, you can see that in the fully mechanized system, we almost doubled the felling productivity compared to the semi-mechanized system. This was mainly due to the higher felling time of the manual system, of course. But you can also see that in the fully mechanized system was less sensitive to the variation in diameter of the trees, of course, in this range. This is not a general rule. About the processing, we have a similar trend, even though the machine was not completely optimized for this kind of operation, in particular for the branching of broadleaves. And even more, the machine was a bit old, so the harvesting head was not very sharp, and sometimes it required extra time that in some cases maybe biased our results. But we had this trend at the end of our study. About the trafficked area, we found that the fully mechanized system had a higher contact pressure because basically the machine is heavier and it has wheels instead of trails, but a lower trafficked areas because the operations were more standardized and concentrated all along one area instead of driving all along the field. These are the results from the penetration resistance survey. This is the percentage of variation from the transect, between the transect and the control area. So, and we compared a before and after situation. You can see some local effects. For example, in this first layer of soil, we have a reduction in the penetration resistance from before and after situation. That was because it was due basically to the wheels and the trails of the machines that mixed the surface layer of soil, and then we had some soil displacement and reduction in cohesion of soil. And then we had a localized compaction of soil in the deeper layer, around between 30 and 40 centimeters. We had the compaction, and this is also called in agronomic field plug pan. It's not particularly relevant because it's quite deep, so the majority of processes which are of interest for agricultural cultivation take place in this area. But still, we found this effect that we have to take into account, especially when we move this to the forest where you don't have tillage and other procedures that can restore somehow the soil. And these are the results from the bulk density analysis. I can just show you some structural support that was given probably by the rot system of the trees. This is again the before and after operation, and this was the poplar road, this was the drainage ditch that was on one side, and here we took the bulk density sample. So this effect, we were actually not expecting this effect, but it was quite interesting and deserves probably more investigations, because this is strongly connected to the bearing capacity of soil. And for those who study bearing capacity of soil, this is actually a very important element. So now we come to a broader topic, which is, as we saw in the agroforestry, but also in general in sustainable forest operations, the production, the productivity, and the ecological process cannot be addressed separately. This is basically the definition also of sustainability. We also add the social point of view. That means that the productivity and the machine traffic are strongly connected. That's why in a next step of my PhD project, we are developing a system, a model basically for soil trafficability by mixing many parameters, especially they are tailored for soil sensitive sites. And this is like the step which I am working right now. I'm trying to build a cost surface map by, as I said before, comparing different parameters. And we got machine data with data loggers. Actually, right now, maybe he's done. My professor is collecting those data from the machine after the operations. And we want to compare, thanks to path analysis and least cost options, we want to compare the baseline approach, which is the traditional approach that is currently used, and the soil-aware approach, that is something that tries to optimize the machine trails. And we want to quantify and find the best compromise so that it can be used in the practical way for next forest planning, forest operation planning. And this is basically all by myself. Thank you very much for your attention.
Thank you very much, Nico. Now, next speaker, Konstantinos Philantos. Next speaker, okay, is coming. Thank you, the floor is yours.
Okay. I suppose you hear me, yeah. Hello, my name is Konstantinos Philidas, short Kostis, I know it's big. So I'm a PhD candidate in the Agricultural University of Athens. I major in entomology. So today I'm not gonna talk about trees, I'm gonna talk about forest pests, a forest pest specific, a really popular one in Europe and in America. So let's get some context first. So it's part of a really large insect family called the Rebidae in the order Lepidoptera, butterflies. So what's really interesting here is that it's a really large family that contains around 25,000 species, described species. There are really much more, many more. And the really also interesting fact is that we identify them based on colors in adults, in like males, females specifically. So the pest that I'm going to be talking today is Limandria dispar, this cute butterfly. So it's a really important forest pest because it attacks, it infests many forest species in Europe, Eurasia, specifically in Greece. This study that I'm going to be talking about was conducted in southern Greece. So some facts about this insect is that it has sexual dimorphism. So the females are the white ones and the males are the brown, gray, boring ones. And we identify them based on color in our field work. So it's a really destructive pest in America because it's invasive. It was brought, I think, 2 decades ago or something. And so it has also become a problem because it has also been infesting trees that are agronomically important, like prunus species. It has been categorized as an invasive in the 100 worst invasive species worldwide, and in many countries, according to the EPO, is a really important quarantine pest. So, some important info about its life cycle and why we will focus on that is because the main damage from this insect occurs around May till July. That's when larvae or caterpillars emerge from the eggs, and that's when they have like insatiable hunger, and they feast on trees from many forest species, oaks, maples, and birches. In Greece, we have many Quercus species. Yeah. Before we go to the trees. That's how we identify them in the field. That's the egg clusters. They look hairy. That's how females lay their eggs. So they lay their eggs in clusters, in trees, on barks, and then they cover them with hair from their abdomen, and that's for protection. And that's how you identify larvae in the field, because they have like red and blue dots in I think the third instar and later. The first instar larvae are much more like dull and black, and that's where the first instar, in fact, is the most important from a public health perspective, because that's the state, the life stage part where they cause many allergies to people, like the pine tree caterpillar. I don't know if you're familiar. Yeah, so they have hair, and that's the allergens, many allergens. I don't know if you're allergic. So that's a typical landscape in Greece that has experienced an outbreak from this larvae. In southern Greece, there are many endemic species of Quercus, like the ilex, I think, and Coccifera. And that's how they defoliate the tree and the shrub, and they cause too much damage, even in like agronomic practices around the forest, because these landscapes are usually agroforestry ones. They're not, I don't know what's continued. They're usually mixed. They have like plains where like livestock feed. They also have like plantations and stuff. So yeah, they cause problems to crops as well. So the aim of the study and the context, basically was the mating disruption methodology that is being used, that has been extensively used to fight many other species of moths and mainly moths, because that's how it works. I will explain. So we use the pheromone that is species specific to its pest, to its insect, I guess. And we use it we have dispensers, so we use them in a more natural way, like drops, like we did in the study. And we place them in like selected areas and around the time that the males fly, because in this species, only the males fly. Females just hang around in a tree and they wait for the males. So we place the pheromone in like a specific formulation on branches around the height they fly, and we expect the males to be confused, and they don't follow the actual females. They follow the scent of the pheromone we placed. So they don't mate, they don't copulate, so they don't produce eggs. So there are no generation. So the areas we selected were in southern western Greece, in the Peloponnese area. We were informed around 2022 that there was a huge outbreak. The pictures from before were from that outbreak, and they had defoliated a huge part of a really ancient oak forest. And the local forestry management was really concerned, and we tried to do some monitoring, and we indeed found that it was a huge outbreak. So we selected two areas, one control and the other where the MD was applied. we selected 4 blocks from each area, and then we made 2 circles, like 10 radius each, and we selected these in order to count the egg clusters, because egg clusters were visible on tree barks. And then we placed the traps for the male monitoring around the circles. And so, yeah, the crosses are the traps. The circles are where we essentially recorded the egg clusters. And that's the, yeah, so its area was around 4,700 square meters. So it was a really large area. And it was a slopey area. It was not like a plain field. So it was kind of hard to reach. So these were the traps, and these are males trapped. We used some on the far right picture. We used the insecticide, this sticker, I guess, because we had to. We were not there. We were not staying there because this study was 3 years long. So we couldn't be there because it's really far from Athens, from the university. So we had to ensure that the insects would stay in, so we used an insecticide agent. Yeah, so inside the blocks, the 4 blocks, in the MD area, we applied 1 drop of the pheromone. That's how we dispersed the pheromone. We didn't use a machine or something. So that's my colleague Maria there, applying the gel. So yeah, he had about 3.3% of this specific sex pheromone that females produce, and we placed them on every tree in the block. So then eggs were counted. We had like weekly records of the traps for male captured, and we had these results about the traps. It was for the males, the monitoring of the female flight. In the first year, in the first summer, because male flight activity is prevalent in the summer, and egg clusters are prevalent after August till May. So the summers were for, I don't know why it's moving. So the summers were for males and the winters were for egg clusters. Here we have the male results, how many were captured. Because the MT application was really late in the 2022 year, summer, it was around the end of July, 31st. So we had a really low levels of males captured, but in the next years, because the application of the MD gel was really early in the before or in the start of male flight activity, we see that the red dots, which are the MD application site results, are really low in comparison to the green ones, which are the controls. So yeah, I think it worked. So in egg cluster recording terms, in the first winter, we had like a really big number for egg clusters we counted inside the circles. But in the next winters, we see a really significant drop in the egg clusters. So we concluded that it worked. It actually worked in these areas. Yeah. So we saw that over 3 years, the last 2 years actually, the male captures and egg clusters dropped, they decreased. The population density decreased, so it worked. And another important factor is the timing of the application. We cannot place a pheromone gel for mating confusion or mating disruption in winter because there are no males to fly, so it will be costly without a reason. So pheromone-based MD is really species-specific, and in forest systems we cannot use insecticides and other active agents, of course. So it's really species-specific. It's really important to note that it doesn't mess with other insects, insect fauna ecology. So it's a really nice way to, an environmentally friendly way to deal with forest pests that actually do a lot of damage in crops as well. So yeah. So we concluded that it is the results support integration of this application in sustainable ways of controlling this pest. And more details are published in this specific study. Yeah, that's it.
Thank you very much. Nice presentation.
Thank you.
And so now the last speaker, Costa Durița, whose presentation deals with the management of oak forest in Serbia. Thank you very much. The floor is yours.
I don't know how to use this. Hello, my name is Costa, and today I'm going to talk about diameter growth of sessile oak as a base for defining forest management objectives. For the first, I need to say the sessile oak is one of the most important tree species in the Europe because of his long lifespan and high quality wood. Also, he is really had a good economic valuables. For this study, we chose area of National Park Fruška Gora because in that time I was working there, and we chose 3 sample plots in 3 different elevations and 3 different ages. But there is no any influence on elevation or age on these results that we made. First, we need to collecting sample cores from the trees. We use only dominant trees, comparing 39 dominant trees from 3 samples. Then we use Dendros, Q-recorder, and Kofesha for dating, cross-dating, synchronization, and other things. After that, we made the structure of the sample plots, age structure, diameter structure, height structure. And then we export that data into file it's suitable for work in R and RStudio and Positron, and then real analysis set started. Then I modeled prediction growth of diameter with Chapman-Richards function, and you can see the formulas for current diameter and the mean diameter. What is the main goal? This is the part of new forest management guidelines in Serbia. Maybe like they are in use from 2021, maybe 2023, I'm not sure. But we have a lot of difficulty with it because they are really confusing. For the example, we have three productivity classify site. We have good one, medium, and poor one. You need to classify stand by, based on the dominant height in that area, in that stand, and it's really confusing. I don't know how to this. This is dominant height for good, and this is for medium. You are really confusing. This is for poor. As you can see, the results, we have a problem to classify this sample plot 1. Sample plot 3 was classified as good productivity. Sample plot 2 is classified as medium productivity, but sample plot 1 is a big problem. Because of that, we based our classification on target diameter. Target diameter is when the most of the dominant trees hit that diameter, then we start reconstruction of the forest. We start cutting the best one, then other things. If you can see in a diameter structure, no trees, there is no one trees who hit 50 centimeter diameter for the best ones. There is no trees like that. So SP2, we also classify that sample plot like medium productivity site, and this is reconstructed diameter increment from empirical data. And as you can see, the biggest diameter reaches SP1, and the growth is strong in early phases of the life and not that strong in the older stages. The same you can see with modeled data, modeled prediction, but model prediction no hit 40 neither in 110 years when you need to cut in guidelines. Guidelines say you need to cut in 110 years, but as you can see, no one hit 40 centimeter diameter. This is, you can see similar pattern of growth in the comparison of modeled data and reconstruction. Strong growth in young ages and not that strong in older ones. You can see that in mean diameter growth. And all I can say for conclusion is that we have really big problems with our guidelines. Not only in this type, I forgot to mention, all sample plots are in the same management type, 26, 21. And we have a problem with height classification, with target diameters, with years of reconstruction and years of cutting. There, in the text of guidelines, you can read that reconstruction needs to start in 70 years or 19. It's a long space between that years. But in the table I show you, you can see the productivity period is minimum 19 years. So we need more guidelines more based on objective and well-defined numerical indicators of growth and productivity across the site of different quality. Not only, I think maybe guidelines is based like only on the good sites. But for these sites, we cannot reach that diameter targets. So we maybe in Serbia we need to recalibrating target diameters accordingly represent accordingly empirical data for realistic and sustainable management of sessile oak forest, and we maybe need more productivity classification. We have more stands for that, maybe, and I'm not sure. Maybe it's all. And the last one. And thank you for your attention.
Thank you very much, Costa. Very nice presentation. I start another time with a good news. All the speakers were very on time and even faster, so we have a lot of time for discussion. So I invite again Aurora, the first speaker, and I invite the audience for doing questions to her.
Okay.
Any question? No one? So, if there is no question for Aurora, I can do one. I have one. But
Like a girl.
it's more a curiosity. Consider that you did your period abroad of the PhD in Poland. What do you think should be changed, if something should be changed, to adapt this decision support system developed in Italy to Polish conditions?
We, of course, made some adaptation of this methodology to the Polish forest contest, because I'm thinking about, for example, changing to adapt the reclassification table with the suitability scores, because of course here the context is completely different. And for example, in Italy we assign, for example, the cable yarder system the very high level, the maximum score of suitability in the such condition where the slope is higher to 60% of percentage. And here in Poland, of course, the majority of the area is flat. So if we apply directly this course, some system can be penalized. But a lot of studies now are suggesting to assign to use cable yarder in the wet area, in wet condition of soil, and very sensitive. So maybe we can rethink to the criteria and add criteria like depth water index can signalize this sensitive soil, or also criteria like the extracted timber amount here in Poland is not so useful because of the continuous cover forest that is applying in Poland. So if we express this criteria in the meter cubic per hectare is not representative, and maybe we should find another way to think this criteria.
Okay. Thank you very much. Other questions? No? Okay, so Nicolò, is your time. Question to Nicolò? One there, okay.
Hello. Can I?
Yes, of course.
How was the spacing between rows and the trees inside the rows? Did you have something like 656, 6x6?
Between the rows?
Yes, between the rows and the trees inside the rows.
The rows were spaced 35 meters. Okay. The trees were around 6 meters.
That's normal. In Serbia, we also plant 6, but we don't have any in between rows. We also have 6 meters.
Okay.
Do you think that have any influence on the your research? That spacing?
The spacing between the rows?
Yes,
probably
between the rows.
Because the rows were quite far. But of course, the spacing between the trees was very relevant. It was very relevant because I don't know how is the space between trees was relevant because As I was saying before, there is some structural support from the rot systems. And then, of course, in the agroforestry context, the spacing between trees is a very important parameter because it also influences the productivity of the agricultural side because of the shade and also the nutritional support for the cultivation. So it's a parameter that it's almost always very carefully tailored.
Other questions? If not, I have one.
Of course.
No, I was wondering, considering that obviously the harvester was more productive, but the excavator-based let's say processing and help of the chainsaw led to lower compaction thanks to the trucks in comparison to the tires. So maybe the best solution should be to mount a felling head on the truck excavator and do all the operation with only one machine, which extract. Maybe just as a suggestion for the studies.
Yeah, and in our case, we had two machines that probably both of them were not very optimized for this kind of operations, but in general, in the tradition of poplar cultivation and probably in that area, they were a bit skeptical. They are not used to use forest harvester in the area because of many reasons, like because of tradition, because of cost, probably. And they have sometimes very efficient teams of people going with the chainsaw and cutting. But we also, we had in our outcomes this evidence that if you can find the best configuration for the machine, then you can get the best from mechanization. And yeah, there was, in that case, you could reduce the trafficked area and you could also reduce the ground pressure. So.
Thank you. Other questions? If not, next speaker, change.
I think I've done something to the PowerPoint. It's moving, but it's okay.
No, there's like a pause. Oh, here we go.
Yeah.
Guess it's this.
Are we ready?
Yeah,
Okay.
because it was moving.
Questions? Okay.
You mentioned that Limantria dispar is destructive and invasive in the Americas. I wonder if it's native in the your study area and what could be like the potential ecological effects of it in the local ecology or the local population of the species itself.
Yeah, so there are there is one species, there are 3 described subspecies. The first one is the Limandria dispar. It's the native one in Europe, the one that it's doing the damage in European forests. I think the Eurasian is called the dispar. I don't remember the.
Limandria dispar asiatica. Limandria dispar japonica.
Yeah, it's the Japan subspecies. There are 3 recorded subspecies. The one we studied was the dispar subspecies. It's a native Lepidopteran species. The thing with this moth is that it has cycles of outbreak. So every around 8 to 10 years, they do a huge outbreak. They destroy trees, and then the populations, due to the lack of food, they drop down. But in that specific area, the outbreak was lasting like many years. So we had to act. I think the focus was to protect the effort to protect the oak trees because they're a really important part of the landscape there due to livestock production, due to agricultural production there, because locals really depend on the trees there. So they have pastures under oak trees. There are agroforestry systems. We haven't done the study on how like dealing with the outbreak would conclude on other fauna, but that's another interesting I think, study to conduct.
Yeah, thank you. And have you looked at what could be because some studies have suggested that there could be long-term effects on species in terms of their behavioral changes when you introduce mating disruption interventions. So have you tried to look on what could be the long-term genetic consequences of this for the species that you are studying?
Yeah, so the purpose of this, I would like to give a context. The purpose of this study was to evaluate on how the MD application would on the results of this MD application, because it has never been done before in Southern East Europe. So the moth was doing damage, but no one really cared about. So we understand that it's a native part of local entomofauna, but when you you're weighing options there, do you do something and help balance the ecosystem, or you just leave the pest destroy the forest, and in order to not do work in a forest system? And that's how we weighed in that we need to act with other local organizations. And yeah, I don't think there has been done any studies on genetic consequences of this MD application. But the thing was, we had to control the moth. We didn't care about its genetic pool. Yeah.
Thank you.
The study was not relevant to the genetics.
Other question?
Okay, very nice presentation.
Thank you. I was a bit fast, but okay.
I have just one question, easy question. How many generations this species has within 1 year in your area, and if there's any concern related to climate change? For example, for the bar beetles in the alpine area, which is at the edge of climatic zone, now there is a concern that the bar beetles are having 2 generations or more, because the warm season is longer. And I don't know, it's maybe in your area it's more stable.
Yeah. So
stable trend.
yeah, we didn't include climate change factors in the study, but the thing is, this subspecies, I think it's found here as well. It's native, but you understand that they're insects. So further down we go, the life cycle is really dependent on the temperature. So in southern Peloponnese, the climate is really dry and hot in the summer. So in 1 year, we really conducted the study depending on the male flight. Male flight starts in differently each year, depending on the temperature. So climate change would probably shorten, I think, the lifespan. But the thing is, they move towards spring, as hotter we get. So they would do much more damage to trees growing in spring. And this damage is irreversible to some trees. So that's how we acted in this area. There was also some outbreaks in the Attica region, where the university is located, but there was a huge wildfire that year, so yeah, it dealt with that. It dealt with that.
Other questions? Okay.
Yeah. So yeah, I didn't answer you. It has one generation. Oh. Yeah. Sorry, got carried away. Sorry.
So thank you for your very interesting presentation. I wanted to ask, kind of out of curiosity, because I study plant protection. Are there any other ways of managing the pest used? Do you know that they are using any
these pests,
bacillus or something?
Other means of...
Other means
okay, so what we consider the location. If it's an orchard, we can spray insecticide. They usually spray insecticide, okay, because it's the most easiest for the producer, I think, and most producers really depend on insecticides in Greece. They usually, in Lepidopteran moth species, in pest species, they use this method because it's a more organic way to deal if their products are organic. But if we are talking about a forest system, we cannot intervene with insecticides. It's not allowed in Greece, at least. So you have to use like predators, entomopathogenic fungi, or like MD. Yeah, MDs, I think it's one of the most successful and easiest ways to deal with it, because you would have to spray fungi, I guess, in the whole forest, and that doesn't ensure that it will succeed. So yeah, I think we have concluded that MD is like one of the most appropriate ways to deal with this pest in outbreaks. We usually don't deal with that pest when it's like existing in forests.
Okay, thank you.
Thank you.
Further questions? If not, thank you. Next speaker.
I'm not ready for your questions, Francesco.
No. Later. First of all, let's see if there is someone from the audience. Some questions? Any questions? Nothing? Are you sure? Okay.
Maybe one easy for start before Francesco's.
No, apart from joking. No. I actually have a question, but it's more a curiosity. No, really. I like very much your study and your approach to redefine the forest management in Serbia. But just I have really big curiosity about your definition of Kopis forest, because you spoke about Kopis, am I right?
Yes.
But you show 90 or 100 years of rotation and diameters from 40 centimeters to above, which, for what I know about Kopis, being from Italy, I know Kopis pretty well. These are not the rotation time and the diameter. Maybe there is some difference in the definition between Italy and Serbia.
Okay. First, I don't know how to say in English our definition, because our name for them, because I don't find anything. Croatian girl is not here. Maybe she will explain better. But after cutting the trees, we don't. In the history after wars, people need to get money quickly, and they are cutting woods.
Mm-hmm.
And after that, we don't plant anything. We don't put seeds in the soil or something. But the things, they are the roots or
the stump.
The stump. That woods is growing from the stump.
Yes, this is Kopis, but at 90 years old, you have resprouting.
What?
At 90 years old, the stump of 90 years is resprouting.
I don't understand the last word.
If you cut a 90 years old oak.
No.
No.
The wait. This is mine.
The last one, yeah.
You can see full name. The aims.
I cannot see. You need to.
I need to. Okay. You see, the main goal of our guidelines is to transform Kopis species in high forests.
Okay, now it's clear. Okay.
Yes, but that we have a problem with diameter. We have a problem with plus trees. I don't know future trees. We have future trees in high forests, but we don't have a future tree in Kopis, because they are not the one.
Okay.
We have plus trees.
This is a pretty complex topic. Maybe we will speak later. But however, very nice presentation.
Okay, thank you. Anyone
Something
other?
more? No? We have a lot of time. Okay.
Oh no, the referee guy.
Because we have the same problem with definition in Poland with Scotch pine forest for for a lot of people there are plantations. Yeah, Scotch Pinus sylvestris.
Okay.
For most of people there are plantations, and for us they are native forests. So it's the same
We
problem
have
with
native
definition.
forest of them in Serbia too. This is not only type this is not only management type we have a problem. We have a problem with almost all types, but this is the type I was studied. But we have a type in Serbia with Pinus sylvestris.
No, this is it's just the consequence of establishing the things from the desk without knowing the, let's say, actually the forest conditions. So people that are doing the policies are not skilled, let's say, are not very conscious of what is the situation in the field, and this creates this, how to say, this difficulties for managers in shaping the decisions. It's typical of different countries, not only Serbia.
It's same thing. Then you have same confusion, or right? I'm right? Okay.
Anything more?
Hey, after you do the hole in the tree, do you do some kind of treatment so they don't get like fungus inside, or the tree doesn't get killed for something like this?
Can you repeat from the start? I don't hear you first words.
Okay. Did you show us that at the beginning of the experiment, you take some kind of wood from the tree, like you poke a hole in it, and then...
Yes, you think this?
Yeah,
with
the age
press
structure.
borer?
Yeah, you take some piece of the tree, right?
Yes. We put some thing, I don't know how to say in English, in that hole. But in my next study, in my master thesis, we used the increment course from pedunculate oak. And because of that, you mentioned, we dig in a really on I think 30 centimeters, not breast height. We dig on 30 centimeters because of that problem. But we always put something in that hole.
Okay.
So cannot be damaged or something. We have a problem also with when tree is already damaged. When we dig, we cannot pull out, or I don't know how to say. We have a lot of problem with that.
Okay, thank you very much.
Indeed, I always told that it's better to cut the tree if you have to measure the age. You just cut, you count the, the tree there. It's easier.
Yes, but for my master thesis, we are cutting 55 trees.
I'm joking, obviously.
And it's like 800 course to send and to measure and to do all.
Thank you very much. So I'm pleased to close the session. We have more time for coffee and discussion. I invite you to continue to ask further questions, further curiosities to all the speakers, and to have a fruitful discussion. Thank you very much.
So welcome on the working. Welcome you on the final part of the session. At the beginning, I would like from the small announcement concerning tomorrow morning, because you have to know that tomorrow you will have a breakfast at 7 o'clock, and after you have to meet before the entrance to the hotel at 8 and 10, and in 5 minutes later at 8 and 15 you will go to the institute to be there at 9 o'clock. It is quite not very far from here, but you need much more than half hour to get. So be on time tomorrow and enjoy your visit in our institute. So I'm a moderator of this session. My name is Teresa Chazubska-Przybył, and I would like to ask the first presenter, the first speaker, to show the presentation. It is Maria Bokuvala, sorry. She is from the agriculture of University in Athens, in Greece, and she will show us the topic concerning the evaluation of trunk and adhesive barrier traps as sustainable management strategies for Taumatopea pithiocampa larvae.
Great.
Okay?
Thank you. Thank you so much.
Thank you.
Good afternoon. I am Maria Bokuvala, and I'm going to present you the study where we have conducted in Attica related to the larvae of Taumatopea pithiocampa. To begin with, I wanted to introduce you to this insect pest. Taumatopea pithiocampa, it's a great defoliator of Pinus halepensis species, and also mainly in Pinaceae, in trees of Pinaceae family. It is we can find this pest in forest environments, in urban and also in suburban ecosystems. Due to the heavy defoliation that they occur in the trees, in pine trees. As a result, trees can die, or especially when secondary infestations follows the defoliation from Taumatopea pithiocampa, such as wood boring insect or fungi. And in the cases that the tree survives, it survives the infestation. There are some issues regarding the development of the tree. So here is you can see the life cycle of the insect. It is a holometabolous insect. That means it passes through complete metamorphosis. These are the stages of its life cycle: the eggs, larvae, and pupae, and the adult stage. In detail, we find the larvae are active during winter. These larvae are inside the nest. They created the nest. And then in February, the last instar larvae leave the trees and the nest. And then In spring, they find they find the soil in order the specific soil, the ideal soil in order to pupate. And then in July, during July and August, we have the adult emergence and mating occurs. And then between August and November, females lay around 150 to 300 eggs in the needles of the pine trees. And then the young larvae start to feed on the needles of the pine trees. Then they create the nests. And finally, the life cycle is continue the next year. So here are the eggs. Females, as I told you, these are the eggs. They lay the eggs around the needles, and then they cover the eggs with scales from their abdomens in order to protect the eggs. The next life stage is the larva stage. They passes through 5 instars. So this is the first instar, where, as you can see, they are completely different than the larvae, the late instar larvae. These are, the color is green to yellow, and they do not have the hairs. They are not covered with the hair, so they are not so dangerous in that stage. And they start feeding. This is the second instar larvae. As you can see, they become darker, and they start making these webs in order to protect themselves. And in this way, they start to create their nests. So here, the third instar larvae is a crucial life stage because they have started. They have the urticating hairs. Where these hairs are very dangerous for human and the pets, because they irritate the skin. When they come in contact with the skin, they irritate, and also they cause irritations. And as you can see, they construct they are very close to finalize the construction of their nests. And here are the fourth instars. And finally, here you can see the nest in the trees and the defoliated tree with the nest. Finally, these are the fifth instar larvae, where they leave the nest and go to the soil downstairs. They create this procession, and they try to find the appropriate soil in order to pupate. And here, these are the pupae. These spots are, in these spots we can find the pupae. And pupation takes place at around 5 to 25 centimeters below the soil. And finally, the adults are moths. So, as I have already told you, the third instar larvae start the third instar larvae is a crucial stage of this species because they create irritation to the skin. Here it's me after a field. So here are some other kinds of irritations. Even after the termination of this stage, and during the pupation or during the summer, there are also, if there are hairs have remained in the environment, they are still active, and they can cause irritations even when they are not in that stage. Here is the larva. So these are they are covered with the hairs, and these are the hairs that are responsible for all these irritations and also to the pets. As you can see, dogs are curious, so they approach the larvae and try to touch. And this is the result after this communication. And here is the latest expansion of this pest. Here it's mainly Thaumetopepite ocamba was mainly distributed around the Mediterranean, but due to the climate change, we can find this pest in northern environments, in northern countries. Concerning the control measures against the larvae, the most common and old measure is to cut the nest, as you can see here, and also the application of the chemical insecticides, but they are not desirable, especially in urban environments. So the aim of this study was to investigate, to evaluate the efficacy of trunk traps, barriers, and also the second year we investigated, we compared a prototype, a trunk trap, and a commercial. The experiments took place in Attica, in Ktima Sigrou, which is a recreational area. It's a park, and people visit this area, as you can see here, every day. And it is approximately a 65-hectare area with Pinus halepensis. We use this commercial trunk trap, which is consisted of 3 parts. The first is the barrier. The second is the downspout, where the larvae passes, and finally goes to the collection bag. And we try to investigate the efficacy of this handmade trunk barrier. It's a sticky surface. We used a piece of bag material, and we cover it with entomological glue. And we put it above the trunk trap, the commercial trunk trap, and below, as you can see, we had 2 treatments. The first one, we put this sticky surface above the commercial trunk trap, and below the trunk trap. We selected 10 trees. In 5 trees, we put the sticky surface above the trunks, and the other 5 trees, in the other 5 trees, we put below the commercial trap. And the sticky barriers were put 50 centimeters above or below the commercial traps. And between the 2 years, the 2-year trials, we selected different trees. And here is the first is the bag, the collection bag. In the first check day, where after we couldn't remove the bag, so the only solution was to cut the edge of the bag and count and remove the larvae. We took them in the laboratory and count them. And then we close the bag with a stapler. So here are some pictures with the traps the sticky barrier, the commercial, the sticky surface with the larvae, stuck larvae in the surface. And here are some larvae in the barrier of the commercial trap. Oh, I have. Yes, okay, sorry. And also, so in the next year, we compare the commercial trap, trunk trap with this prototype, which is consisted of the same parts, but from different materials. Some pictures are there here, you can see. And we follow the same procedure. Here it's me. I try to protect myself during the observations. And so we move to the results. Concerning the captures in the sticky surfaces below the trunk, the numbers were very low. So in 2015, we captured 5 larvae, and 8 in the next year were in the concerning the traps, the commercial and sticky traps above the sticky surfaces above the commercial. These are the numbers. And here you can see that the sticky surfaces did not capture so many larvae as commercial did in the first year. Here you can see the start of the emergence of the larvae. These are the results for 2016. And here are the comparison between the commercial and the prototype trunk traps. As you can see, there are no significant differences. They worked equally. And to conclude with, so here you can see that when larvae stuck in the surface, the larvae that follows can move above them so they can pass this sticky surface. That's why we observed lower numbers of captured larvae. And also, since both the prototype and the commercial had equal number of captures, we suppose that the opening was the downspout. Since they have a similar downspout, this helped the larvae to go through this the downspout inside the control bag. It was easier to apply to install the traps, the commercial traps, and this project was supported by this study was supported by the Life PISA project. There are the results we can find in Journal for Economic Entomology, and thank you, and I apologize. From Croatia, and she will be talking about the strike weaknesses, opportunities and threats in Kórnik forest management, insight from the Croatian. Sorry.
Hello, everybody. My name is Lucia. I am coming from Croatian Forest Research Institute, working in the Department of Silviculture. And today I'm going to present a research that we did within our department. And it's a topic that is kind of important, but also middle ground, gray area that nobody actually knows what to do with. And that's coppice forest management. So starting from the background, as I said, coppice management is one of the most complex issues, not only in Croatia, but in Europe as well. There, in Croatia, most of copices are European beach copices, and they are under office branch Gospić. That's Lika County in Croatia. It's a mountainous continental region. You can see it here. In the small picture, it's the borders of the county, and on the right picture, green is pinpointing the office administration branch. Yes, management of those copices are shaped by specific historical, economic, and social circumstances. By historical, we mean a war that was happening over a couple of years, and also different rules, governance was happening. So it was different management approaches that left actually copices neglected. Also economic overexploitation and social circumstances over a couple decades, actually depopulation happened. Also natural site constraints like harsh terrain and heightened climate change exposures, increasing climate extremes further complicated management. Objectives through our research were many. Evaluate whether natural regeneration can be used more effectively to reduce planting costs and improve biological efficiency. Also to assess the density, vitality, and spatial distribution of natural regeneration as a basis for conversion or substitution. Identify the most appropriate substitution approaches, whether it will be direct, indirect, or mixed. By direct, it means in one step to substitute beach copices, indirect in couple of steps through planting or sowing, and mixed to combine clear cuttings on small areas and substitution under the cover, forest cover. Also to align the selected method with site productivity, species composition, and the current structure of the coppice stand. To identify risks related to the emergence of competing or invasive vegetation. This is also in context of climate changes that invasive species are increasing. To assess the likelihood of habitat degradation, including soil disturbance, erosion, or loss of humus. And to integrate mitigation also in context with climate change and prevention measures into the official state management plan. We conducted SWOT analysis and identified major strengths, weaknesses, opportunities, and threats. There were many more in the actual report, but I chose the most significant nowadays. Major strengths, definitely sustainable forest management and SC certification, high biodiversity and increase of diversity species, and natural tree species composition, shorter rotation periods that enable more biomass production and economic returns, high ecosystem service value and proven ecosystem service values, carbon sequestration potential, cost-effective regeneration, and experienced use of mechanized harvesting systems in challenging terrain. Major weaknesses would be high costs and also low economic profitability of those operations, difficult terrain and inappropriate or non-existing mechanization, low accessibility, limited infrastructure, degraded coppice conditions with weak regeneration and declining biological potential, complex and slow conversion or substitution of stands requiring high expertise, insufficient knowledge transfer, data availability and strategic planning tools, and increasing exposure to climate change and other environmental pressures. On the other hand, we also identified major opportunities, and that's improved carbon sequestration and compliance with climate obligations, long-term improvement on productivity, timber quality and stand stability through silviculture treatments, substitution of coppice stands into, which is in our research the most important aspect, and that's higher value forest forms with improved natural regeneration, development of modern data systems such as UAV and other cameras, technologies and science practice cooperation, cross-sectoral cooperation also, growing demand for biomass and wood products within the EU bioeconomy and circular economy, and modernization of forest operations supported by EU funds. And major threats, climate change impacts as one of the most significant, also lack of skilled labor, which is in Croatia in the last years shown increasing. There is no much of skilled labor. Market instability and low economic viability of coppice forest investments, weak local economy and continued depopulation pressures that are happening over a couple of decades, increasing costs and reduced access to funding, biological risks and uncertain long-term forest stability, including carbon stock loss, complex substitution processes under increasing biotic stress, more pests, more diseases, invasive species. And also, as our previous presentation was about Pithiocampa, Tomatopia pithiocampa, it's also is spreading in Croatia through continent further. Yeah, difficult terrain with erosion risks. So to conclude all of our SWOT analysis, in Croatia there is total of coppice area in Lika region or Gospić forest branch is 77,140 hectares, of which only 2% of that area is under active silvicultural measures, such as thinning, tending, site preparation, planting, sowing. We figured that the shorter rotation periods would be better, especially for beech. Satellite data improved forest monitoring, and that's with UAV sense and other sensing, modern contemporary sensing. We still concluded that coppice management is highly complex, and therefore we picked the WO strategy as it's some kind of middle ground strategy to reduce weaknesses and use opportunities. Substitution to high forests is recommended to climatogenous forests, heterogeneous. And thinning improves growth, quality, and conversion potential. And to conclude, I would like to mention a project that is led by Croatian Forest Research Institute, and it's about it's LabAdapt. It's establishment and development of laboratory for adapted forest reproductive material to endure climate changes. And yeah, if anybody is interested in the project, you can check our website, and you have also emails on the slide. Thank you.
Thank you, Lucia, for keeping of the time. And now I would like to invite another speaker. It is the Petr Zapletal from Mendel University in Brno from Czechia. The floor is yours.
Thank you. Hello everyone, my name is Petr Zapletal and I'm a PhD student at the Department of Geology and Soil Science of the Faculty of Forestry and Wood Technology of Mendel University in Brno. Today I would like to tell you something about different regeneration pathways of implementation of silver fir into Norway spruce monocultures with a focus on the soil, mostly on the nutrients in soil. So for starters, I would like to introduce the problematics connected to silver fir in Czech Republic. As you can see, nowadays, Czech Republic is covered by 33% by forests, and 45% of the 33% are covered just by Norway spruce, Picea abies. The problem is that most of these Norway spruce monocultures are unnative to the sites. The sites are not very good for this Norway spruce, and nowadays we have seen a huge dieback of Norway spruce monocultures in Czech Republic, because it used to be 53% coverage just a few years ago, and we are losing huge areas of forests because of the incorrect sites of Norway spruce. On the other hand, we currently only have 1% of silver fir compared to natural state that would be 19%, which is more than naturally Norway spruce would be. And also it is recommended that we should have at least 7% of silver fir in Czech Republic. in the past, just a few centuries ago, in the 17th century, over 30% of Czech forests were covered by silver fir. But nowadays, as I've said, we have reached only 1%. The main problem was the air pollution, because silver fir has a big problem, especially with sulfuric oxides. And we have seen a huge decrease in vitality of the silver fir stands, and also inappropriate forest management in terms of mostly selective cutting, because silver fir is a great species for construction wood, and it was selectively cut in the past. So we have lost a lot of the silver fir stands. But there is still a huge potential for the silver fir. But as we have heard already, the temperature in Czech Republic has increased, the average annual temperature has increased by 2 degrees Celsius already. So the temperature rise itself is not a problem for silver fir, because many manuscripts show us that silver fir even increased its increment thanks to the increment of the temperature. But the problem is that also the evapotranspiration is increasing. And if the site does not have enough water, the vitality of the silver fir is decreasing, and thereby it is not able to regenerate itself anymore. So that's why during our project we are focusing on different management regeneration pathways to see which ones actually suit the silver fir best. Here are a few pictures of silver fir and why it is an important species and why we should focus on it. First of all, it is a great species for diversifying of forests, because it is a highly shade-tolerant species. It is able to occupy even just small stand gaps, and it is also able to thrive in those gaps for many decades. Even 70 years under full canopy is not a problem for silver fir. Also, it is very wind resistant. As you can see, the surrounding trees were wind thrown, and the silver fir remained as the only species on the sites. It is also able to live on clear cuts, and as I've said before, it is a great species for construction wood. But a huge problem nowadays in Czech Republic is that even when there is at least some natural regeneration of silver fir, it is preferred by the wild game, by deer, and it is very hard for us to maintain natural regeneration without fencing it, because we have overpopulation of wild game in Czech, so that's a huge problem nowadays. So now to the project and study itself. For this study, I'm presenting some of our results from very different pathways of reintroduction of silver fir into Norway spruce stands, which are natural regeneration on a small-scale clear-cut and underplanting the silver fir under the Norway spruce stands. It is important to say that after the underplanting, a huge dieback of the Norway spruce stand happened. So this is the underplanted variant. All of the forest around had to be cut because it died because of severe droughts. So this actually nowadays looks like more extreme conditions than the ones on the clear cut before, because it was just a small scale clear cut. But I will show you the data about soil. And it's important to say that both of the stands are at the 5th forest vegetation zone, which we call Abies fagus in Czech Republic, because it's the only one that has Abies in its name, because it's optimal for silver fir in the Czech Republic. Here you can see the sites where they are located. And now to the methodology. First, we have collected organic horizons using this sampling circle, because we know the area of the circle, so we can later quantify the amount of the topsoil or the or the organic horizons per hectare. And for the mineral soil, we used soil cores because then we can calculate the bulk density and later calculate the stocks of the nutrients in the mineral soil. This is the design of the sampling plots. Each sampling plot was divided into 3 subplots, and each subplot has 4 sampling points. So we wanted to really represent the whole stand and to even show the variability of the sampling points. So I think that, and also I forgot to say that in each sampling point, 3 samples of mineral soil were taken, and then they were mixed together, like the 1s together, 2s together, and 3s together. Now to the results. So talking about the forest floor, I said that it looks like the underplanted variant now looks like it's more extreme, but actually even after the dieback of the forests around, it still remained a lot of humic horizon. Actually, it's more than 10 times more than in the clear cut. And what is important to say with the clear cut is that the humic horizon was not even present in 1/3 of the sampling points. There was also a huge variability in the clear cut topsoil compared to the underplanted variant, where it was actually pretty similar to each other. Here you can also see shallow soil profiles in order for us to be able to classify the humus forms on each sampling point. So here is a clear cut. You can see that there's almost no litter and just a very thin humic horizon compared to the underplanted variant. Now to the results of the stocks of nutrients. The highest difference was found talking about phosphorus, which is very often the limiting nutrient in forest stands. And we have found much higher stocks under the underplanted variant compared to clear cut. And it also there was a huge variability in the clear cut, showing us that the underplanted variant has more stable nutrient dynamics and also more favorable for the trees. Base cations, they showed us similar results, much higher stocks in the underplanted compared to partial cutting, and the differences were found in both mineral soil layers. I'm sorry, I forgot to say that the mineral soil was sampled in 2 different depths. One was 0 to 10 centimeters, and the second one was 20 to 30 centimeters, because 0 to 10 is often called the topsoil, and we wanted also to see whether there are differences in a deeper mineral soil, and that's why we have chosen the 20 to 30 centimeters. So, as I've said, even the base cations showed us huge differences between the variants. All of the nutrients were more present in the underplanted variant, and also the base saturation in the topsoil in the 0 to 10 was much higher under the underplanted variant. Now to the soil reaction, or pH, if you may, and aluminum. Interestingly, the underplanted showed higher concentration of aluminum, but what is important to say is that when you divide it, if you divide the base cations to aluminum, the underplanted variant showed much better results than the clear cut, even though the concentration alone was higher in underplanted. So it's important that we should not conclude the data just by the concentration themselves, but to see how what are the ratios between the nutrients. So now, just for the conclusions and the discussion, clear-cut was associated with much lesser organic horizon accumulation, which doesn't necessarily mean that there are more nutrients in mineral soil. As I've showed before, actually, even though there are more, there is much higher accumulation of organic horizons in underplanted. There are even more nutrients in the mineral soil. So overall, we can say that the underplanted variant was much better to remain the nutrients and to stabilize the nutrients in the soil. Also, it's important to say that the silver fir trees remained in a very good vitality even after the dieback of the surrounding stand in the underplanted variant. I am only presenting the results of soil research, but there are colleagues from ecophysiology, also from forest protection, and colleagues focused on mycorrhiza. So I don't have all the results here, but as I've said, the vitality was actually really good in the underplanted variant. But the problem is that because of the surrounding forest was cut, the silver fir show severe damage caused by early frosts. Because there's nothing around, so the frost was much more severe than in the covered variant with surrounding stand. And also, what is important is that the higher nutrient stocks were found in both mineral soil depths, not just in the topsoil, but also in the deeper mineral soil. And as I've said, even though the concentrations of aluminum were higher in the underplanted variant, more favorable ratio was found there, so the concentration alone does not mean that the conditions are worse. So we may say that the underplanting variant is better than planting on the clear cut. Of course, this is all based just on one-to-one stand, because we don't have the results of a laboratory analysis on another stands yet, but there will be more results, and actually manuscript is in preparation, so soon hopefully you will be able to read better results. Thank you very much for your attention.
Perfect speech. And now the next speaker will be Irina Gyubilai, from University in Tuscia, in Italy.
Yes.
And she will talk on the topic.
Yeah. Hello, good afternoon, everyone. I'm a researcher at University of Tuscia in Italy, and I'm working in plant pathology. So today I will present the evidence of the plant pathogens, emerging plant pathogens in the forest ecosystems in central Italy, and discuss how the climate pressure is reshaping the host microbiome interactions. So you are probably all familiar with the Via storm, which in 2018 caused a massive forest damage in northern Italy, over 42,000 hectares of forest affected, and millions of cubic meters of timber on the ground. Events like these are becoming more frequent, but the climate pressure is not only about extreme events, because in general the temperature are rising, especially in the Mediterranean area, and at the same time the precipitation is decreasing, and as a result the forests are increasingly under stress. Under these conditions, trees behave differently, and the forest ecosystems are pushed out of the balance. Under low climate pressure, in general, the forests are in balance, and the ecosystems are resilient. As climate pressure increases, we have that the forest health start to decline with tree stress, reduced defense, and lower resilience. In these conditions, we have the pathogens gain advantage with faster development and new host interactions. And when both processes occur together, forests may cross threshold leading to decline with higher mortality, disease outbreaks, and canopy loss. But what are we actually, what is it happening in our forest, sorry. What is happening? We are observing an emergence of new pathogens, new host pathogens interaction, and increase in pathogen virulence. So during my studies, we identified emerging pathogens on forest species, track disease dynamics across seasons, and explore hidden microbial reservoirs using a metal barcoding approach. So let's start with emerging pathogens. So in 2023, we identified Colletoticum fiorine for the first time on Fagus sylvatica in Italy. Colletoticum fiorine, it's a fungus that causes the anthracnose symptoms on leaves, and it's an emerging generalist pathogen, so it doesn't have a specific host, but can affect different plant species, so forest species, ornamental, horticulture, and so on. But our first report is not only one in Italy, because in the recent years, the Colletoticum has been reported, for example, on hazelnut, on olive, but also on walnut. So this suggests a rapid expansion of its host range, probably like due to more favorable conditions, such as warmer temperature, for example. A similar pattern was observed for Neophysicoccum parvum, which we reported for the first time on Ailanthus altissima, an invasive plant species. This is an aggressive canker pathogen causing bark necrosis, defoliation, wood discoloration, and also plant health, of course. Ailanthus altissima is an invasive host, and for this reason can be a source of the pathogen. So at the same time, Neophysicoccum parvum could be used for biological control for the Ailanthus, but this requires caution, because also Neophysicoccum parvum is a generalist pathogen, so that can infect many different hosts. So here we can see the recent, our first report on Neophysicoccum, but our record on Ailanthus is not only one, because it's been reported also on hazelnut, mango, and almond. Both Colletotricum and Neophysicoccum parvum reported, probably there are not isolated cases, but part of a broader emerging pattern. So let's go now to the field observation. In autumn 2023, we observed a high incidence of foliar lesion in Monteraschio beech forest, a natural beech forest that is very important and very particular for its altitude, which is one of the lowest recorded for Fagus sylvatica species. From the symptomatic leaves, we identified many different fungal genera in the same stand forest using the culture-based isolation and Sanger sequencing. Most of the genera identified that we can see on the graphs are opportunistic pathogens that appear when the trees are under stress. For example, we can see Alternaria, Colletotricum, Biscognauxia, and so on. But what happens when no symptoms are visible? Many microorganisms are already present inside the plant tissue without causing any visible symptoms because leave as endophytes forming in the communities within the plant, and they can become pathogenic when the environmental conditions change over the time. And to investigate this hidden component, we analyze the fungal and bacterial and endophilic communities in asymptomatic beech Fagus leaves, and using a metabarcoding approach. And we found that these communities are not static, of course, but change over the time during the growing season. Here we analyze the fungal communities and we can see that they change over the time, and some potentially pathogenic genera are already without visible symptoms. For example, we have Apionemonia, Ramularia, and Biscognauxia, and as the season progresses, also the stress increases over the time, for example, due to drought or warmer or so on. And some pathogens like Ramularia, for example, increase over the time. At the same pattern was observed for the bacterial community. Their composition also changes over the growing season. This confirms that the trees host a dynamic and complex microbial community, which can be a hidden reservoir of potential pathogens. So let's go to the take-home messages. These findings are based on observations collected during my PhD that had a different thematic. So for this observation, I don't have the correlated climatic variables analysis for this observation. But however, they clearly suggest that the climate changes is reshaping the plant microbiome interaction, and that the new pathogens are emerging in forest ecosystems in central Italy. So in the future, it will be important to better link the climate variables with the disease dynamics, for example, by developing the predicting models. And at the same time, we need a proactive monitoring strategies, including, for example, molecular tools like the metabarcoding approach to detect the pathogens early, even before symptoms appear. This is particularly important for forest management under changing environmental conditions. So thank you for your attention.
Thank you, Elvina. And now I would like welcome the last speaker during the session. It is Monika Sołoniewicz from the Warsaw University of Life Science from Poland, and she will be talking about the mechanical variability of Scotch pine wood from different provenances. So Monika, the floor is yours.
Thank you very much. Hello everyone, I'm Monika Sołoniewicz. I'm PhD student in Institute of Wood Science and Furniture of Warsaw University of Life Sciences. And today I will be talking about the wood itself, so maybe something different than we were talking before. I hope you have the energy for the one last presentation. So without further ado, let's start. Here is the content of the presentation. At the beginning, I will talk about the project that the work is a part of, then the classical material methods and the results. So for the start, as I said, my work is a part of quite a big project called Dendrospec. We're working, my Institute of Wood Science and Furniture from Warsaw University of Life Sciences with Institute of Forest Scientists. Also, we are working with Institute of Dendrology of the Polish Academy of Sciences in Kórnik, and with Institute from Slovenia in Renew. So the acronym is Dendrospec, but the full topic of the project is spectroscopic methods for Scott spine dendrometric features and wood properties characterization, reflecting its provenance and genetic variation. You can see on the graph here, it's representing the scope of the research because we have quite a lot of topics. You can see we basically combine dendrometry, physics and mechanics, chemistry with genetics, and also near-infrared spectroscopy and hyperspectral imaging. So that's why there's a lot of people working on the project, because we are trying to get as much information from the wood and the trees that we can. So today I'll be talking about the work package 2, the physics and mechanics. And it also would not be the whole research, just the tiny part, because it is really a lot of data and we are still in the process of analyzing it. So about the materials. In the 1960s, the seeds from 16 different populations in Poland were taken, and the seedlings were planted in Rogów, here marked it 1, in central Poland. So all of the trees that we are testing were grown in the same environment. And we are trying to see the differences between the different parental stands. And today I will be talking about the homegrown ones from Rogów, so here, from west of Poland in Gubin, and also from one stand in mountains from Nowy Targ. And the material for the mechanical and physical testing were obtained from basal part of the trunks below the breast height, and we took samples from pith to the bark in north and south direction. So the samples were standard for the mechanical testing, 20 by 20 by 300 millimeters. And today I'll be just talking about the density, static bending strength, and also modulus of elasticity. We did a lot of different testing, but to get the 15 minutes that I have, it will be only about the 3 mentioned properties. So about the density. Here you can see 3 graphs. Here is for the homegrown ones from Rogów. Here is the from the west side, 11 from Gubin, and here is the mountain population. You can see from the first glance that, maybe I will see what it is. It's the average values of the samples here from the pith to the bark. It's like the N1, 2, and so forth, from the north direction and the south direction. So you can see that in each graph, the density of the wood was lower in the pith and higher in the older mature wood. That is not new knowledge. Everyone from wood technology knows that. That the juvenile wood is less dense, especially in the coniferous trees from plantations. But we can see that the trees from Nowy Targ had lower density overall. So the mountain population they were as dense as the ones from different parts of Poland. There we can see, in the case of bending strength, there is also the difference between the juvenile wood and the outer mature wood, but you can see that the difference is much bigger. So we can see that the strength of the wood is not correlated one-to-one with its density, but it's also something else. It could be the anatomy, also the fiber structure, because in the juvenile wood, the fiber are pretty much, they are shorter, the microfiber angle is different, so all in all, they are just less strong. And you can see, when we are talking in the project about it, we call it like the smiley faces. So you can see in the case of the homegrown ones and the one in from west side of Poland, they were much more happy than the mountain region. So the mountain trees were not as happy to grow in central Poland than the different populations. You can also see here, from the homegrown ones had the bending strength of almost 150 megapascals, but from the mountains it was 83.5 megapascals. So that's quite a lot of difference. And we are in the process of analyzing all of the 16 different populations. And we are also getting the genetic information. I can talk about it. I do not have slides for that in this presentation, but we can talk about it later if someone is interested in that. And here is next for the modulus of elasticity. It's pretty much the same as the bending strength. The populations from the central Poland and from the west, Gubin, they performed better than the one from mountains. It is, I can say there is, it was consistent that we see that the trees from 16, the origin 16 from Nowy Targ, the mountain trees, they were just like, I don't want to say worse, but they had less density, they had lower strength in compression, bending, they were less hard. So pretty much in every tested characteristic, the origin from Nowy Targ was like the outlier, they were like different. It was pretty clear to see. And also in the chemistry guys that were like doing the analysis of the chemical properties, the content of the cellulose, lignin, terpenes, and all of the different analyses, the 16 was really different. So to conclude, the studies properties of the examined wood shows a recurring variation at breast height of the trunks in the north-south direction, and the highest average values of those presented here. The highest density and MOH, like the modulus of elasticity, were obtained for the Gubin origin, so the one from the west side of Poland. The highest average value of modulus of rupture, the bending strength, was obtained from Rogów origin, and the lowest studied properties were from Nowy Targ. And like the message is that it is possible to identify the phenotypic variability of Scots pine from different provenances. And we can use that information to determine seed material that can be used to ensure efficient stands in the future. So there is contact for the project manager, and thank you for your time. If you have any more question, feel free to ask. I'm happy to talk about the project because it's really great research, but yeah, it's so huge that I can talk only about the small part in here in the presentation. So thank you very much.
You obtained, and now we have 7 minutes to the discussion. So we can start now, and we have half hour to discuss. So half hour, yeah. So maybe we can start from the last speaker, from Monica. If you have any questions, so you can ask. Yes. Yes, please.
Monica, how do you think if we plant the same trees, not in the Rogów, but in the Nowy Targ, this Nowy Targ provenance could cope better?
Yeah, I do think that the trees from the parental stands, they could be better in their native environment. That's the next part of the research. We are planning to do the next project to test the trees that were in the parental stands.
So we can say that the results from this project they just confirm the relevance of the provenances of the seeds. That we should keep going with the provenances.
Yeah, I do think that we should keep going to the provenances. The trees I like. I can talk about it later because we got the genetic information and it is kind of different than we expected. Sadly, we do not have the genetic information from the mountain region. This is really a bummer because we are trying to understand why they're so different. But hopefully in the next project we can get the genetic information about it, and then we will see what's next.
First, thank you for the presentation. I have a question. If Nowy Targ was the only one place from mountains from where you have samplings, or there were more samplings from mountains?
No, in case of the mountain representation, it was only Nowy Targ. So yeah, only Nowy Targ in this project, but maybe in the next.
So maybe you have an idea if the properties can be caused from this, that it's from mountains and other area. If you will have samplings from other point in mountains, it can be similar. Do you have an idea? What do you think?
Yeah, if the parental stands are kind of the same as the Nowy Targ, yeah, I think it could be similar.
And are you sure that this mountain population is native to this place? Because you analyze the most variable population like Bory Tucholskie, Supra and so on, where the Scotch pine is in the optimal conditions, but my question is, on what elevation grows this Scotch pine in mountains? Because it's probably not native there.
Sadly, I could not give you the information because the seeds were taken in the 1960s, like 40 years before I was born. So I'm sorry, I cannot give you that information, but maybe if I talk about it with my project manager, he will have the information for you.
Maybe that's the reason that you don't have information about genetics, because they are from a completely different place from Europe, for example.
No, about the genetics, it was some different problems. So it's not about that. It's they didn't want to say that it's not native. It just different problems. So yeah.
Thank you. I wanted to ask you whether any of your colleagues were focused on the ecophysiology as well, or if you only look at the wood properties, or like if there are any other colleagues looking at other parameters than just wood.
So yeah, you can see that from the work package 1, all of the dendrometry. The forest guy were about all of the properties of the trees itself, and people from Kórnik, I think they were like about the needles and all the different stuff that I do not know about.
Another question to the Monica, or we can ask speaker. So, let's go to the Irina. Yeah. Have you any questions to Irina on the forest pathogens, on the dynamic?
Yes, thank you. So I've dealt quite a bit with similar metabarcoding stuff, mostly on birch trees, and we take a very healthy, nice-looking leaf and sequence the fungi, and we also see all kind of alternaria, ramularia, and whatever else pathogenic fungi growing there, and the same applies, for example, to our ash trees, which have a lot of Hymenopsiphus fraxineus growing on them, and sometimes also on non-symptomatic trees. And it really puzzles us. Can you really tell when a presence of pathogenic fungus actually becomes a disease?
I don't know if I understand the question. So when I can see the symptoms of the pathogens during the season, it's a question.
Are you able to, for example, model when tell just by DNA data when the presence turns into a disease?
Okay. Yeah, now we don't have the models to predict the development of the pathogens during the seasons for now. So this is a project can be development in the future. This analysis, for this analysis we use the asymptomatic tissue, and probably we can see any symptoms at the end of the seasons, when the plants are more stressed, for example, for increased temperatures, or in autumns. So, but I don't know if it's clear.
Looks like it isn't. That was what I was wondering about.
Any questions to Irina? So the next speaker was the Petro. So maybe you have questions. Yeah.
First of all, congratulations for the presentation, the interesting results. Only one question, I'm wondering, which is the distance between the two sites? Because as far as understood, you have one site in one part of Czech Republic and one site in the other part of Czech Republic. How much is the distance, and how you check that these sites were really comparable, apart from the climatic zone? But how you can be sure that your results are not influenced from by soil management histories and a lot of different things, because this can be some point that in the case of of paper writing, the reviewer can pick up and tell, but why you don't have, you understand what I mean? This can be picked up, so.
Yeah, I fully understand what you mean. That's also why I'm very open about it, that it is just single stand right now. They are pretty far from each other, but that was actually what we intended, because we did not want to sample all of the, there are actually 6 variants, and we have also 6 variants on 2 different site types. So there will be 12 variants, and each variant will have at least 2 stands. So we did want to avoid sampling just, let's say, one geographical region, because then you cannot generally say that this is the better one, because then you are just on one, let's say, the same bedrock, same locality, and there's an effect of a small locality. So we wanted to sample across the Bohemian Massif, you know. So right now we only have the results from those stands, and we are waiting for another replicates. The samples are at the laboratory right now. That's what I said at the end. So yes, I understand this, but it's actually interesting that talking about the bedrock, the underplanted site is on let's say less fertile bedrock, and also the bulk density was smaller in the underplanted. So that would mean that actually if it was biased by bedrock, then underplanted should have lesser stocks. So this is even more interesting, that on let's say worse bedrock and in worse conditions, we have better results on the underplanted, which looks like really the underplanted is much better, because there's no, the soil is not uncovered, you know, it stays covered, so there is no rapid decomposition of the organic material, and there are no losses, or much smaller losses than compared to the clear cutting. But yeah, I fully understand what you mean, and that's why there will be more replicates, but they are not analyzed yet, so.
Okay, just because probably for this I was telling that you have so interesting results, also clear results. But you know, then when you go to the scientific part, you start the data analysis, modeling, etc., someone can pick this up and question. Instead, so if you plan, as you are doing, everything since the beginning, this can really, how to say, make an impact of your study. So this was just a suggestion somehow.
Yeah, and sorry, I forgot to answer the first part of your question. We chosen all the stands by the Norway spruce had to be 100% represented. It had to be of the same age before the silver fir was planted there. The silver fir had to be of the same age. As you could see, it was 15 and 13 years old. Yeah, the difference is minimal. And also, we have in Czech Republic, there's like a forest typology, which focuses on the site conditions in terms of herbal layer, also the fertility of the soil, and it says what would be on the site naturally. So we selected the stands so they are comparable by the forest typology. So that means that we are trying to do the groups, and as I've said, there will be 2 site conditions, meaning one is water. One of the groups and the stands that I have been presenting today are from a group of non-affected by water, and then there will be another variant. All of the variants will be studied on sites that are affected by water. So we can then recommend where you should use which way to reintroduce the silver fir in order to keep the best vitality or keep the nutrients and so on and so on. That's why the project team is so big, and there are like experts from different fields. So we want to really create recommendations, because we really need to reintroduce the silver fir to Czech
Perfect.
forest.
Thank you.
And what about carbon and nitrogen?
Yeah, I have not presented the results here because I do not have all the data on the top floor, on the litter. But talking about the mineral soil, there are no significant differences, which means that, as I've been presenting, there are like 90 tons per hectare of humic horizon in the underplanted. So there will be much higher stock of carbon and nitrogen in underplanted variant because there are still 90 tons of humic horizon. But talking just about the mineral soil, there is no significant difference right now.
So the next speaker was our colleague from Croatia, Lucia. Do you have any questions to her?
It's not my fault, sorry. No. I think that I have a lot of questions, so maybe we speak later about this.
Okay.
But I have specifically one. When you did the SWOT analysis, in the advantages of, as far as understood, of copies management, you put, among the other things, something like mature mechanized harvesting possibility, something like that. Now I don't remember exactly the slide. So what do you mean with, how to say, that mechanization of corpus harvesting in Croatia is established, or what? Or whatever. I didn't get this point.
I'm not sure I understand the question. Mechanized use of mechanization that is adapted to the terrain.
But in copies, in the transition, in the conversion, in which?
Yes, in any type of work activity in restoration.
Okay, we speak later. Let's say okay.
Okay. Thank you.
I have first, thank you for your presentation. I have a question about the larvae. You say they can irritate the skin, cause there a problem for people living near the pine trees when they spend time there, or it's not a usual problem for people living in such places?
It's a usual problem for people who live or who walk near the larvae, because they feel the fear and they release the hairs, and when they come in touch with the skin, they cause all these irritations. And also I said that this hair can remain in the environment for a long time, so they are active, and even after usually larvae appear during early spring, so you can have allergic irritations during the summer, for instance. I had the problem. I faced this problem during the summer when I checked the traps in that place for the adults.
Thank you. And one more question. Are they native? Is this a native species for your country? And the problem is now bigger than before years, or it's now a problem that they are a lot of the larvae and people have more irritation?
You mean this? If it is a problem nowadays or it used to be, I think it will be a problem, because every year we see the processions of the larvae. And this is a big problem, especially in areas with pine trees, because they infest specific species of Pinaceae family. So it's a big problem, yes.
But it's not a new problem?
No, it's not.
And it's
a
native
European.
species?
This is a problem in Mediterranean basin, in Greece, in Italy, in Spain, in Algeria, Egypt, and around the Mediterranean, in Turkey. And now it has spread in Germany, in France, North France. So yes, it's a problem.
Thank
It
you very
was,
much.
it is, and it will be a problem. It continues to be a problem.
Yes, thank you. I also have a question about larvae. You mentioned several times that they walk in processions, but could you explain why?
They create this procession.
Yes.
Because there is a leader. Usually it is the first caterpillar, the first larva, is the leader of the family, because all larvae in the nest are siblings. So this one is a woman, is a lady, is a female, a future female, and she leads all the family in the appropriate soil in order to pupate. So that's why they create this. The first one is the leader, and the next they follow. The next larvae follow the leader of the family. And if someone remove the first one for a few minutes, they stop this procession. They stop walking. And then the second one become the leader of the family.
So about the hairs, it's like there's something on them, like the substance, or like why they're so irritating?
The reason of the irritation? You ask the reason of the irritation?
Yeah.
Okay. They have. They activate the histamine of the skin. So they have a kind of toxins. They are covered with a kind of toxins. So when they come in contact with the skin, they activate the histamine of the skin and that's why we need the antihistaminic pills during the period. And that's why they irritate.
Okay, and is there any other species that does the similar thing, or like it's like this one that's all of the Mediterranean? Because I was in Slovenia and saw like this weird nests on the pines, but I didn't know what it was.
Yes, there is Thaumetopeia wilkinsoni. There are some other species, but in the Mediterranean and especially in Greece, we suffer a lot from Thaumetopeia pithiocampa in the whole Greece.
Okay, so like...
And especially in urban environment, in schools, because we have so many pine trees. So during spring, you can see all kids red.
Okay, so like the different species also like the irritating, yeah?
Yes.
Caterpillars. So I didn't go to them when I was in Slovenia. Okay, thank you very much.
You're welcome.
I want to ask you, do they have no natural predator? Because they are pretty vulnerable when they are walking in such a long line. So is there nothing that actually predates on them?
Yes, but not in that stage. If I remember well, not in that stage. I'm not sure. But at the adult stage, some birds are predate them.
So do you think that this might be a way of natural control to somehow build nests or whatever to for the birds, or like do you see anything else than the mechanical barrier to protect the trees?
The trees. Apart from that traps that we tested. In that project, we also examined mating disruption as a way to control the species. Our aim was to decrease the number of the caterpillars because they create all these health issues. And also we had examined there are traps for the capture to capture the adult. So we we tested different type of traps, and all these are published. If you are interested, I can tell you later the publication.
Thank you.
You're welcome. So, yes, another one.
Just a curiosity, why do you need to develop, to use a homemade trap solution? Because are too expensive, the commercial one, maybe?
Yes, it is used to, Marlon, it was used to apply, to put this kind of trap. And also it's cheapest than the commercial trap, but it didn't work. And so the next step was to create, to develop, to design a prototype with cheap materials and recyclable material. That's why the next year we compared the two traps. You're welcome.
So I think that's time to finish our session. Thank you, our speakers, for a perfect prepared presentations, and also the audience for all questions. And I would like to remember about today's meeting before exit to the hotel at 8 and 10. To go to the institute to be on time at 9. And now we will have a dinner, so welcome and thank you again.